And I'm going to take you to another field which is very, very important. Although all of us are interested in sequencing and to get to know about the DNA, we also know that like the amount of information which is being, it's available to the organism, and to us to understand about the organism's functions are also increasing. Okay, let me start with one of the slides where it's like there is a phenomenon or an effect called street light effect. So it is like when we as students and researchers of microbiological sciences or pathological sciences, we have something in common. We tend to seek the truth where the process of seeking is easy rather than where the truth is. So wherever, also there is a short story where, a mullah who was searching for an item which he has, dropped somewhere, misplaced somewhere, and he was searching under street light, and somebody asked him, why are you doing it here? And they asked, where did you leave your material? Where did you lose your material? Then he said, like, okay, I lost somewhere. Then why are you searching here? He said, this is where the light is. So same story also goes with this, because many of us are trying to understand about the DNA, the information available and packed in DNA of the organism, and try to understand about the function. But our understanding is slowly increasing because the kind of tools which we are introducing into the science is also increasing. Probably the first toy which was used as a scientific tool was the microscope, right? And now many tools are becoming again toys. And we have several important things which are coming up, like from microscopy to nanoscopy, for which 2014 the Nobel Prize was awarded. And we also know about the use of microscopy to understand about the microbial world, and we know about the number of organisms which are available on the tip of organ, which is going to be like, when you can see here, it's about 9.10 to the power 13 of a mass of one bacterium, and you can see how many such organisms could be seen on a head pin. Okay, and then we also mastered the method, like cultivating them, so that we are able to get a lot of information. So now new methods are coming on to understanding of the organism, or also about the health of the microbial cells. And this is one which I want to show you, because they are introducing electrical impedance as a measurement to understand about the cell health. Okay, this was in 2021, and all this unit, the unit is very, very small one. The sensing area is so small one. So that kind of developments are also going. So don't think only about DNA sequencing. So there is a lot of new tools which are coming up in the field to understand about the health, and then also about the functions of different organisms. I'm just giving you an idea, like what are the numbers in terms of numbers. These organisms are about 10 to the power 30 cells on the planet. So if you wanted to know about every one of them, it's going to be really difficult, because they also store about 350 to 550 petagrams of carbon, which is almost equivalent to what is being stored in the plants, the higher plants. And they have about 85 to 130 petagrams of nitrogen, and then also about 9 to 14 petagrams of phosphorus. And then they have an interesting feature, 10 to the power 1.7 into 10 to the power 30 cells per year is having a turnover rate of 6 to 25 days. This was one of the papers which came from William Bernie Wigman from University of Georgia, Athens, whose lab I also happened to visit. Okay, so we wanted to learn about these organisms, and well, we wanted to understand about 10 to the power 30 microbial cells which are present in the earth, which is going to be really difficult. Well, we have to make a humble beginning. This is what done by Professor Francis Kerl from Prague, Czechoslovakia. So what he did was he started having his own personal collection, and that collection was then transferred to Vienna in 1950. That was the first time the culture collections were made, and then it was who made a request to Professor Kerl. Okay, and also at the same time in 1906 there was a culture collection, that is Central Bureau for Sculptures, which was in Bern, Netherlands. In 1905 IRA was established. 1906 these guys have got the cultures of microorganisms. Okay, and now we have several such culture collections across the world, I'm going to take you to some of this information so that you will appreciate like how much of search has to be done before you go for sequencing your organism, also about the genes, because whatever genome, metagenome, whatever it is. Metagenome is a new field, but otherwise we have a good number of cultures which are available. So if you want to know about the genome information of any culture, you probably should go through most of this information so that you don't have to spend money again for the information which has been already collected. So that is one of the toughest job for us. So how many of you start going to the library? All of you, I think, because we don't find like IRA library is outdated. So now have all information in our internet. So you go to many of these journal websites or journal databases, and then you search for information. These cultures, several information available in the form of in the World Wide Web. So you have to better undergo, see all of them to understand what are available and what are not available. Okay, and the permanent office for this culture collection, World Federation of Culture Collection, is there in Belgium, right? And those information I'm not going to give you, but you have two. There are two important information. One is about World Federation of Culture Collection. The other one is about International Union of Microbiological Sciences. These two have a good number of cultures available. And then, as I told you, there are several members. WFCC members, we called it, World Federation of Culture Collection members, and who are all members from India. So we have National Chemical Laboratory has got a national collection of industrial microorganisms, who is also a member of WFCC. And then we have National Cultural Collection of Pathogenic Fungi, which is from ICMR, and it is there in Chandigarh. And we have National Fungal Culture Collection of India, which is again from Agarkar Research Institute in Pune. And we have Indian Type Culture Collection from IRI, Catalog of Fungal and Bacterial Culture, which was released in 2019 by Division of Plant Pathology. And also we have MTCC, Chandigarh, which has got a good collections available in India. These are all members of World Federation of Culture Collections. So if you really want an authenticated culture, you need to approach them and collect those cultures, okay? And then we also have WCC Merson World Data Center for Microorganisms. There's again another body which is having about all the information related to the culture collections specifically for microorganisms. They have several such databases. We have CC Info, Global Catalog of Microorganism, CGM Type Strain Genome Database, Analyzer of Bioresource Citations, Reference Strain Catalog, and we have GC Meta Microbiome Research Platform, which has been recently added. So I'm going to go through all of them to give you an information so that you should be aware that where are the places where you can look for cultures and also get to know about what kind of cultures they have. So we have Culture Collections Information Worldwide, CC Info, and which has got several such collections, culture collections of 831, also covering about 78 countries and regions, and having microorganism 3 into 10 to the power 6 plus. That many organisms are available in these culture collections. They have bacteria 1.4 into 10 to the power 6 numbers of million organisms are there. Okay, and so this culture collection worldwide is a database management system for culture collections across the world, and it includes CC Info and Strain. It's a world directory of all registered culture collections. Database Strain includes list of holdings from registered culture collection. Actually, it started by Professor Skerman and his colleagues in University of Queensland in Australia. From there, it has become a FCC World Data Center for Microorganisms. So he published the World Directory of Collection of Culture Collections, Cultures of Microorganism, World Directory in 1972. From a personal effort, it has become a world body now. Right. And also we have CC Info Strains, where you can get to know about the strains which have been identified and authenticated, and also you can search from these databases. So this culture collection information worldwide has got all the databases related to the countries, and the cultures which have been submitted by each one of them, and the number of organisms belonging to bacteria, fungi, yeast, protozoa, human cDNA, and many such cultures which are available in this. And we have another database which is called Global Catalog of Microorganisms. Again, it has got a good number of strains which is running into 67,000 and then odd organisms which have been listed, and where this, and also we have an article which has been published in BMC Genomics about the Global Catalog of Microorganisms, a comprehensive database and information retrieval, analysis, visualization system for microbial resources. So these are available to us, and you can go through this paper to understand about like what are available in Global Catalog of Microorganisms. Okay, so they have a different, they have some particular definition for the culture collection, Global Catalog of Microorganisms, also the motivation and the references which are listed in this website, right? And it is giving you the kind of cultures which are coming from different countries, what are the culture collection centers, and the number of cultures which are from different countries, and then all those information which are available. Okay, maintaining these databases are not that easy because you need to know about the bioinformatics tools. Not only that, like computer analytical mathematical tools are required to understand how to prepare a database, and then to make them available easily for the users. If you really want to search everything from a primary journal, it's going to be really a tough job. Even if you have them, like okay, all the information which have been published in primary journals are in the databases like NCBI. All of them have been taken out and then they have been curated, and these informations are available in these databases. So, and this gives you like what are the organisms which have been preserved from around the world, and also they have a standards, and then what kind of what you call from different sources or the habitats from which these organisms have been collected. All these informations are available, and source of organism, like what are the organism, and then different sources like soil, and then water. All those informations are available in the number of. So all these things keep changing every year. Every time when the culture gets in, you have a database, numbers keep increasing. So you can always find out whether someone has done these collections and put it in the database. That's what makes things easy for us. Otherwise, as I said at the beginning, you will be spending money again on projects on an organism for which the information is already available. You don't have to repeat them. So this also gives you about the geographic origin of strains, and about 5 million strains, and then most of them they come from US, and also China is now taking over. And since you can see several of these databases are now available with experts in China, right? And we have another one, and a consortium called Asian Consortium for the Conservation of Sustainable Use of Microbial Resources. And again, 3 such culture collections like MCC, MTCC, a national agriculturally important microbial culture collection of ICR is also listed in this. So it is now the consortium is having all the culture collections, and 3 such culture collections are now from India. Okay, then we have another database called Type Strains Genome Database, which is also giving you information about different phyla and how many of them are. The strain genome information is available like in different phyla, and this is also another important database which has got some information about the project and the kind of data which are available, and search menu, tools, and also statistics. I'll just give you some information about like what is the standard operating protocol for the sample submission of CGM projects, and then there are a couple of references which are available, and again authored by Chinese experts. And you can download the method, SOP, standard operating protocol for submission of genome information. Okay, and also they have all the species information, so which are the names of those organisms are being curated by LPSN. So you just can't give randomly some name or the name which you like for any organism, which has to be approved by the LPSN and also for some of the international journals. And only then they will take those information and then make it available in the database. Right. This LPSN is a list of prokaryotic names with the standing in nomenclature. And now this has been, it has now gone to, the Dutch culture collections, and so that is being more now looking after this. As of 2020, the database regularly augmented LPSN database at DSMZ, and the basis of LPS service. So everything is now to be done. So even if you get a new organism, you cannot, you have to consult these people, and then also like how to use, and then give a new name for your organism. Well, and then they have information about certain tools related to genome analysis, species identification, and manual. So the best thing is, like, once you know there are some databases, spend some time in the website to collect like information related to what are the information which are available in there. It is not just one single, gene information or genome information, which is normally available in a research paper. In a primary paper, you will have just about one organism, maybe about one gene. But if you really want to know about the importance of that particular gene across several organisms, you want to know about several organisms which have been collected from different countries or different habitat, then you have to understand about the databases. Otherwise, your information is like I'll say a drop in the ocean. So you may say that I sequenced a particular gene, but when you go to the databases, you know the gene has been sequenced by several people, or a genome has been for a particular organism is available, and you have several such information can be. Now, what you really have to do is you will have to understand about those information and then come out with the scientific concepts related to that particular gene or the genome, not just sequencing alone. So part of whatever we have been teaching you about the sequencing or about the genome information is only a beginning, okay? And when you want to really know about the scientific principle, you have to move on to the next level and see about the global pattern or their behavior, distribution, biogeography, their functions, everything has to be understood from this. So these things have been made easier because of a collective efforts of every researcher. So if all of you are making contribution to these databases, you just imagine what could be the richness of the information available in the databases. And also we have a responsibility, because if you don't make the right input into the databases, you're going to spoil the database. So again, the common term, garbage in, garbage out, which is being used by bioinformaticians and computer science people, well, is applicable to us also. If you send a wrong sequence, and then if you are not sure about the quality of the sequence or quality of the genome which you have done, and if you upload them, if you give those information available, it is garbage in process, and rest of the people are going to suffer because of committed. Well, and they also have a manual, and wherein they talked about like how to get information on NGS reads and TGS reads. How many of us know about TGS? NGS, well, most of you must be knowing about next generation, okay? TGS, by the time you all mature, this will be an outdated technology. That is third generation sequencing, TGS, which is employing single molecule sequencing, directly sequences individual DNA or RNA strands, and then in a sample with amplification. So this technology is going to be the technology by the time you become a senior researcher. In next 10 years, this will be the technology. So NGS, what we are now teaching, is going to become a foundational thing or a basis on which the whole information or whole understanding of the biological life is being built up, but TGS is going to improve, right? So be prepared for the next technology. So this is the point which I want to say. And they are available with the manual. This is SOP for information which is from NGS read and TGS reads. Well, they have prepared themselves for that. That we have to prepare ourselves for the next stage. Well, they have all information related to RNA and tRNA and many organisms. Everything is available. So don't be, I mean, like you will understand. Okay, what kind of information is available in the databases? They are not available in the library. Hard copies of journals, you don't get many of them. Maybe you will know some references like how to use the databases. I'll also show you which is the journal, which is every year in the month of January, they come out with their articles related to different databases towards the end. So they have a procedure called sequencing novel sequences, sequencing novel species. So they know like, I want to sequence. So if you download all of them, they will give you the steps which have to be followed if you want to do the novel species. So everything related to the types, strains, genome database, right? And we have ABC. ABC is analyzer of bioresource citations. So it is now taking the information from the primary journal, everything related to the culture or about the gene or genome, and having a different database so that you don't have to do the search in the normal journals. Like again, it's a refined database made from all other sources. It's a platform which is being prepared to share all kind of information related to species, strain, culture collections, and then also author, what they call is scholar, institution, culture collection. So if you all those related information of microorganism, you can collect from this ABC tool and services. They have different tools available in the website itself, right? So if you go into the website, they also list the top 20 species is the largest number of articles, something like how many citations of your work. You see, like number of organisms, list of organisms with a large number of citations which are available. So when you go through this website, you will know, okay, whether my organism have been worked on by other researchers. How many of them? Is it a new one? Or what I'm looking at is already available there. All those information you can see from this website. And of course, this is to give you a workflow, mining workflow, ABC mining workflow. So they go from patent, journals, genome, nucleotide sequences. So what they call is information crawling. Then regular expressions like species name and then they remove all the duplication, remove false positive, then get into the database and then GCML data provider and web service. So all these things are being done. So otherwise, you just imagine you have to go through all the journal and make a database for yourself. Is it possible? See, suppose say I am working on rhizobium. I want to know what are the information available in rhizobium. Even if I do manually the search for the information related to rhizobium, I may retire and get out of this institute. Okay, it's not that easy. So many papers are coming on rhizobium, but I want to know like what others have done. How much information is available? Why should I do such things which have been already done? All those things you can understand from this workflow. Okay, so they have done everything easier for us. So that is what we have to appreciate. The hard work has been done by these researchers and also by the work, database organizers. Okay, so we have got world DNA Center for Microorganism, WDCM reference strain catalog. Again, the strains, number of species, culture collections, and all those things are available in this website. So again, these are, you can know, like, in fact, like, okay, let me get back to this. Here, this is actually related to handbook of culture media for food and water microbiology. Most of the organisms have been isolated from water and food microbiology. Okay, so everything is cultured, and it is not metagenome, but all those which have been collected from those sources are being available here. And we every year some of the strains and the species get into the databases. You have a list of all of them coming up. It's available in the database itself, right? And these are the people who are involved in it. Like this is one from Belgium, Dr. Philippe Desmat from Belgium, David Smith from Cabby, UK, and also this WDCM by Ma, director of WDCM in China. I'm showing China because what is interesting to me is these websites are now having Chinese language. Okay, maybe in next 10 years or 20 years, the language of science is going to be in China, Mandarin. Okay, slowly you may have not only read, not only learn bioinformatics tools, and then you may also have to know Chinese language. Okay, and here it's workflow for RSC processing. So all this being cross-checked. See the control and then kind of entries into different databases. This is what we call them as queuing or the workflow. Okay, all these things are done so that you have the reliable information. So this is the person, Dr. Ma, who is in China and Chinese Academy of Sciences. Now they have a standard operating procedure for World Data Center for Microorganisms, reference strain catalog, released in 2017. And it's also WFCC affiliated culture collection, and which is having strains and all. So numbers, yes, you can always understand, from different affiliated centers. Also, we have, this World Data Center for Microorganism, WDCM, is launched in 1966, and then which is the data center of WFCC and Microbial Resources Centers of Network. MIRSEN was the earlier version, but now these are being, they are together as the center, right? And they have it all started from the University of Queensland, Australia, and now it has become a global effort. And of course, in this website, they also are given what are those resources which have been considered for nomenclature, NCBI taxonomy, species 2000, LPSN, Burgess Manual of Trust. Some of you must have been taught about Burgess Manual of Trust, but now these are also being taking the responsibility of giving the nomenclature to the organism. And they have metagenome from NJGI metagenome portal, EBI metagenomics, and metagenomic submission guide. Everything is available. So if some of you are interested in metagenome, then we have to use this information, and then those who are interested in nomenclature have to go to these sites. Okay, and this is what I was saying. This center, they have the site picture. It's all in Chinese now. All these informations are now coming up slowly in Chinese, right? Well, and these are the actual information related to those CC info and GCM, ABC, and RSC. I'm not going to go through all of them. This is to give you an idea. Databases are several nature, several different types are available. So one has to cross-check what is available in one database and go to the another one, and then also understand what is available. Some of you may be interested in analyzer of bioresource citation. It's a data mining tool, extracting strain-related publications, patents, nucleotide sequences, and genome information. So you don't have to go through the journals. So you go to this website and understand about what you look for. Well, and those who wanted to use the reference strain catalog, go here, right? So these are the information which is also available in nucleic acid. Nucleic acid comes out every year with the articles related to database. So one such is World Data Center for Microorganism and Information Infrastructure to explore and utilize preserved microbial strains worldwide. There they are given how informations are collected from publications, patent, genome, nucleotide sequences, then gets into the ABC. From ABC, how the data retrieval analysis and visualization system is used in GCM, and metadata is available and reference time, right? So I think I also showed you about the workflow, which is also given in the paper itself, how information crawling is done, then going into the database. This is to you, as a researcher, you are supposed to have an idea, because otherwise, you will end up in a world without information, right? And if you don't have the right information, your work is not going to be appreciated, right? And they also have a global catalog of metagenomics, which is now recently made available in this in the website. If you see most of the meta China, right? So the information database is now maintained by Chinese researchers. Well, I think this is to show you like how to pre-process, then the DNA gene sequences, metagenome sequences, and submit all of them, right? So they have also have a culture collection catalog of microorganisms related to what I really wanted to show is not only the type of organism, they also use the sequencing method 454 solid and Illumina ion torrent. So they now specify which method by which you get your data, metagenomic data or genomic data. So now this is going to be important. Like some of you, if you are using a particular technology or platform, you can always check whether somebody has done it and whether you can use it as a reference for refining your information. So by sequencing method, which is very interesting, and also like if the metagenome is available, you can also have an idea like what is the abundance, taxa abundance. So if you have a metagenome and you prepare a taxa abundance pie chart or a bar diagram, and you can easily compare where else you have similar such abundance, which is not easily possible for us. Like if you really want to compare it by yourself, again it is going to take a lot of time, right? Each paper has to be screened, but now everything is available. So this also makes things easy. Like then if you know the taxa abundance, you can find out like what kind of a function there. You can at least make some kind of a predictions using information related to the taxa abundance. Of course, world directory, I mean like now we have the culture collections. Now you just understand like culture is now into 3 into 3.3 into 10 to the power 6. So it's running, several such collections are available. Bacteria, fungi, viruses, and cell lines. So don't think that like, okay, my culture is the culture, right? There are several people who are having good collections, are being done in the past, and those cultures are available. So all you have to do is like how to get information about them. So ABC has got, look, the purpose of ABC is also to to honor something related to the Budapest Treaty on international recognition of deposit of microorganism purpose of patent procedure. Well, some of you would be competitive enough to collect a new organism, try to patent it, and then you will have to see what kind of information, I mean, whether the organism is really novel, has got a novel functions, which you can go for a patenting. Again, this information you need to know from ABC. ABC will makes things easier. So this is Budapest Treaty was signed in 1977, came into force in 1980, and we have 78 countries which are signatories. World Intellectual Property Organization in Geneva. Culture deposited any one of the IDA, International Depository Authority, are recognized by the member state for the purpose of patents. So this is also important. Where do you submit your cultures? If you submit it in a culture which are not recognized as IDA, then you can't claim it as you can't claim for patenting. So you need to know whether you can pay so much of money and then go into this IDA for summation of the cultures, right? So we have 27 culture collections in India which are registered with the WDCM. Some of them like NCC, IMTAC, and National Fungal Culture Collection of India, National Collection of Industrial Microorganisms, NCL Pune, and National Agriculturally Important Microorganisms, and National Collection of Dairy Cultures in Kannada. But all those things keep it aside, but only two cultures in two centers like MCC and MTCC are recognized as IDS. I think there is a news, I mean like last year I came to know like NBM is also considered for ID as an IDA. I'm not sure about it, but let me first, you may go to the website of NBM and then find out whether their culture collection is also coming under IDA. So this is also important for you to understand about the for patenting purposes. And yeah, we have two of those, culture collection which I wanted to show you. MTCC in Chandigarh is very important. accept microorganism for deposit under general, safe, patent, and IDA deposit. Clearly, they have announced it in their website. So if you really want to, submit your culture for patent purpose, you can go there. And also, National Center for Cell Science has got a National Center for Microbial Resource, and which is also accept your culture for patent purpose. MCC accept microorganism for deposit under general IDA. This is the kind of services which are available in addition to accepting your cultures. Flowchart is given. Go to the website and understand what you have to do if you have a new organism which you wanted to submit for patent purposes. So I'm going to take you to another important, should I say, a database, but this is one. It's only only museum of microbes in the center of Amsterdam in Netherlands, which is very, very important place. So if you happen to go to Amsterdam in Netherlands, go to Microbia. This is the only place where we have a museum for microorganisms. So some of you may have fungal collections, fungal collection in our institutes, but if you want to see about, the intention and purpose is also given in the website. I'm not able to read many things from here, but if you go to the website of Microbia, spend some time to learn about the Microbia, the actual motivation and purpose. And if you have an opportunity, visit. In fact, Dr. Raimakers, he showed you a video about how microorganisms are associated with the human being. It's a fun video was available. So the website has got a lot of information about the lab talk, about biotechnology, rise and shine with the microbes, microbes which are present in your toenail, cheese, and belly button, and so many other things. So there are several interesting, information which are, that is about the museum which is available to the public. And all other databases and culture collections, strain collection is meant for professionals like you. Soon you are going to become a professional in dealing with the microorganisms. Also, we have some national collections. I'll say ATCC is important one. Many of you must have heard about American Time Culture Collections, and which is there in Virginia, Manassas in Virginia, under National Institutes of Health. So they got a rich collections. You can always get it like the MTCC we have in Chandigarh. If you really want to get good, before MTCC was established, many cultures from India were sent to ATCC, and they are also available there. But if you really want to have some internationally recognized acclaimed cultures from ATCC, you can pay and collect them. So ATCC has got microorganisms, certified materials, all microbiome standards which can be used. So there is also, there are several such information available in the ATCC genome portal, and visit the website, learn more about it, because it's not that easy to communicate all the information which are available in ATCC in few minutes to you, right? I'm only showing you what is important. So go to the website, learn more about it. When you have time, spend more time, so that you will understand what are the things which are possible you could do with ATCC. Also, we have another database. Again, we have algae base. And so this website has got information about 1,69,000 odd cultures, species of algae, photographs, and then papers and distributional records of everything. What is interesting is this was started by an individual, Michael Gary, who was the founder and director of Algae Base Project, and then in UK he has done it, and I think his wife probably, both are involved in making this culture algae based, and later on now there are several contributors who are personally contributing to the maintenance of this database. That's something which is interesting. Yesterday, I think Bill Gates came, wanted to give money to our institute or many researchers for research, giving money for maintaining databases. That also we should appreciate. Well, it is not one way like, you spend money to give the cultures, but how do they spend money for maintaining them, making available to others? It's also a tough job. And also we have involvement of Phycological Society of America, British Phycological Society, International Phycological Society, Korean Phycological Society, and they have maintained it. Do you find an Indian Phycological Society here? We need to find it, making contribution for keeping such databases globally. Right, and there is another data, like stock center, fungal genetic stock center, and which is available in Department of Plant Pathology, Kansas State University. Okay, so again, universities also having a good collection, like the one which I said about the fungal, like the ITCC, which is available in Department of Pathology here. Like, so they have one, and then there is one called E. coli database collection, and this is just about E. coli. So there are several workers, who concentrate on E. coli alone. So they can go into this, and then they know about like genes, ORF, regulatory region, promoters, terminators, tRNAs, rRNAs, everything about the E. coli strains. They are also available. And of course, all of you must be knowing about the NCBI. NCBI is was founded in 1988, which is largely sponsored by the Senator Clark Pepper, and then it's the United States National Library of Medicine and the branch of NIH. This is available. So many of us have started using NCBI in the beginning because for every gene we go to this website, and we were trying to do a lot of our initial work on this. So this is the real purpose, and what I was I appreciate more is, from an alphabet of only 4 letters representing the chemical subunits of DNA emerges a syntax of life process whose most complex expression is man. So again, we are concerned about ourselves. So from DNA, we wanted to understand more about man. The unraveling and the use of this alphabet to form a new words and phrases is a central focus of the field of molecular biology. The staggering volume of molecular data and its cryptic and subtle patterns have led to an absolute requirement computerized databases and analysis tools. This is the purpose. So even if you call, like you are going to work with the 4 alphabets, or rather 4 letters of a DNA, but unless you have these computerized databases and analysis tools, you don't understand the life process. This they have understood, and to meet the challenge, they have the NCBI. Well, and NCBI has got several information related to gene, and also they have several databases under gene and expression. If you look at all this, chemicals, bioassays, data, software, DNA, RNA, domain structures, genes, expression. So I'm only showing you a couple of them so that you know what is available. So they also have all the databases, download, submission, tools, and how to do. So this is, again, now the other one is genome. So when you go into the genome, they have, again, databases related to the genomes and map. So what kind of a database is available? So this is ever increasing. So every time they come out with the new information about proteins. So GenBank, NCBI Bookshelf, and BLAST, all those we have been using. So we should all appreciate like how to, making things available for many of us. Also, GenBank overview, and the GenBank is now released. 254 version is available, and if you look at it, like the kind of the current release has got 241 into 10 to the power 6 traditional records containing 3, 6, 9, 12, 1.7 into 12 base pairs of sequence data. So this much information is already made available collectively from different researchers across the world. So if you do a sequencing, don't think that you are doing it for the first time. You need to know, yes, this much is already available. So we have so much of sequence data which are available, right? So well, again, it's got GenBank. GenBank also got a block. So now the new information which I like it, they have a block to say, block in the sense relatively informal, not source structure. So you can understand like what is NCBI, who works here, kind of information is something which is really good for us to know. Like otherwise, you only know the few pages of NCBI, but you do not know what is going on behind the NCBI. How do they work, and how people come together to work. So how they work and what kind of databases which are available, and all those things are like, are available in the NCBI Insight blog, something which you should also understand. So, well, I'm not going to go through this. So we have ribosomal RNA summation vision, an entrance system. So it's an integrated database retrieval system that provides diverse databases at the 2.5 billion records. So we have a query and all those things you can understand from NCBI. Blast updates, quick blast, smart blast. And then there is also an article which is getting published regularly in Nucleic Acid about database resources for the National Center for Biotechnology Information. So Eric says, so he lists us like what kind of records which are available, literature, genomes, genes, clinical nature, protein, and chemicals. Just imagine the number which are being, the information which are stored in NCBI. You need to go through this article to appreciate how much hard work, how much information are made available for others to use. Equally, there is another center which is important to all of us is EMBL and EBI. So European Molecular Biology Laboratory, European Bioinformatics Institute. So they have an important website. Not only NCBI, you also go to this website to know what is going on in the Europe. So this is, I think about 27 member states are available in EMBL. So they are responsible for having this particular website and an institute which is there again in Oxford, Cambridgeshire, in Wellcome Genome Campus. So they have EMBL. So what is more important is this EMBL, EBI has got a training events. So every time they list what are those which are available as webinars, course, you can attend them. So go to this website. So what we are doing currently is a workshop to teach you about that. But these people, this website, and they are teaching you several such courses which you can register and attend. Okay, and also give you an opportunity to apply for a position in EMBL and EBI. So this website, if you visit regularly, you can learn how to go there and work, and how also to get an employment in EBI, okay, and EMBL. So this is very recruitment procedure, careers, international applicants. Go there and visit all of them. So this is to give you kind of an information like what is available and all. It supports life science research throughout the world by providing open data. They don't claim that it is only for Europe. It is for the whole world. Open data, open source software, analytical tools, technical infrastructures. So many tools are about them. So Magnify and then Microbiome Analyze and all those things are available. So go to the website and learn. So if you have a lot of time, don't waste it. Go there to the website and learn about them. So they also give you like why there is a need for bioinformatics. Biological data is the bedrock of all life science research. Few examples how it can be used in a beneficial way, understanding plant genomes, cancer, and MRSA, methicillin-resistant Staphylococcus aureus cases. So you can understand. And there is also an important information about the Darwin Tree of Life project, where how phylogeny tree, which yesterday they were teaching you how to prepare, and then what do you call tree phylogeny? Yes, all those things you can understand from there. So what does it matter? Like what is the big data and big demand? They use 155 peta types. So how many tera types? 155 into 10 to the power 3 terabytes, 10 to the power terabytes, 10 to the power 12. So that much of information is available in this. Every weekday, well over 38 million requests are made. You just understand 38 million requests. So that is the amount of information people look for, and they also get from EMBL or EBI. They got a lot of information like same thing like BLAST, nucleotide, protein, HMMR, UniProt, ensemble, data resource. Tools are different, data resources are different, and program access are different, all available in EMBL. So then how do we do data submission? They also give you the protocols. Resources are run as a collaboration with the partners in 16 countries, in which data and metadata are shared that users can search and find resource-specific data relevant to their research through any of the partners in collaboration. So these repositories you can go through and then get them. Also, like how to do research at EMBL EBA. It is not only a repository for all data information, a lot of research. So EMBL research published journals, nearly 40% of the papers in 5% of the journals in biochemistry, genetics and molecular biology. So a PhD program they have, some of you are already a PhD. EBI, we also have DNA data bank of Japan. So DDBJ. So of genetics, Shikoka in Japan. Okay, so they have DNA data bank of Japan has got search analysis, tools and databases, and how do we submit similar functions. Some of them are unique to Japanese collections. So it's also again, although I told you like Chinese is taking over many of these databases, the Japanese also they have their own databases, right? So you can look at the kind of data information which is available. Say into I think the bases are running into billions. So 20 or 30,000 billions sequences are available. So you have information about the bases, sequences for human and all other things. By each organism, like organism and the number of bases related to that organism is available. So you have now choice. Okay, if you don't like NCBI, go to EMBL. If you don't, okay, you want to cross-check with the DDBJ, come to DDBJ and get information, right? So we have, then also they have another database center which is called the International Nucleotide Sequence Database Collaboration. So they have data, all the INSTAC is a long-standing foundational initiative operates between DDBJ, EMBL, EBI, and NCBL INSTA. So this is between covering all of them. So instead of doing it individually, you can go to INSTAC and then start searching for them directly. Okay. So I think I have less time, so I'm going to go through fast. So we have another important website which is called JGI, Joint Genome Institute of Department of Energy, USA. So they have integrated microbial genomes and microbiomes. So they have information again about DNA and RNA sequences, single genomes, archaeal and bacterial, eukaryotic and viral genomes, and genomes from metagenomes. Also available in JGI. And JGI has got, they have information related to annotated DNA, RNA sequences. Well, and also we have features related to, like how different organisms and on which they have information and all those things available, number of bacteria, isolates, max and single genomes, all those things are available. So this, there is a paper in nucleic acid about integrated genome and metagenome comparative data analysis system, and that gives you like what are the genomes available and overall view about JGI, integrated microbial genomes and microbiomes website, right? And we have Gold Genomes Online JGI, which is nothing but Genomes Online Database. Well, I'm going to go through fast. So we have another paper which came in 2023, 25 years of Genomes Online Database, data updates and new features in version. Version is 9, where they inform about the curated resource and then how your input is critical to ensure high quality and accurate metadata in Gold. As I said to you before, out principle. So if you're putting them there, it's going to be a lot of problems. Okay, organization of gold is given, and well, I'm not going to read all these things for you. And there is also some, kind of an information which are required from us. That is known as minimum information about a marker gene sequence, MIMAX, and minimum information about any sequence specification. So this is an important paper to understand like what is expected from you as a researcher if you are trying to use this databases or if you want to submit your information. So this is available in Nature Biotechnology. So read about this, and also they have given you how to make sure that your quality is good enough for submission. So then there is another website which is called Microbes Online, which gives you information about several genomes and gene information, homologs, gene domain families, domain process. So it gets into different aspects than what is available as a culture. So JGI is also has got websites. Go through them. That will be more important. So and then you can do microbial genome database for comparative analysis. It's a new database for a comparative analysis of completely sequenced microbial genomes, the number of which now growing rapidly. The reference paper is in nucleic acid in 2015. So if you want to make comparative analysis, this is very good. Instead of you downloading data from different databases and do that, you can go to this website and do it. Okay. So welcome to MBGD is a new side paper, and then this is what we have been. We started our career with RDP. So this is ribosomal database project, which is maintained by Michigan State University, and still it is okay. We are using it, but of course now that we know like microorganisms are too many, so this reliance is very limited. So they have a functional gene pipeline and repository. So this gives you not only ribosomal, also about the different genes in RDBI. So go to the website and understand about like how to submit, how to manage, and how do you use this database. So we have another one called fungi database, DB, and this is for the fungal organism. Like you have bacteria, so you have here FungiDB. So this is 61 version is released, and again, data mining and functional genomic analysis can be done using FungiDB. And we have European eukaryotic pathogen genomics database resource. UPathDB is just for eukaryotic pathogens. Well, and then we have a genome database collection, which is meant for cycle. This is a database which uses E. coli K12 and MG1665 as a strain, as a reference genome for ecosite, and this is a collection of pathway genome database for model eukaryotic organism and microorganisms. Okay, so you have been taught about KEGG and other things. So if you want to know about the pathways specific for the bacteria or E. coli, you can use again. So this pathway and genome database provides electronic references on the pathways and genomes of many organisms. Well, that's ecosite, and this is exclusively for E. coli. That's what I showed you, pathway biosite to E. coli database. So Metapsych is another one for metabolic pathway database. Then we have BACTIVE, which is an explore bacterial diversity, and this is with the German collections. Okay, they have a dashboard where they have number of strains, species, and the number of organisms which has been available in BACTIVE. BACTIVE is a database structure on taxonomy, morphology, physiology, cultivation, isolation, molecular data of prokaryotes. And again, most of the things are obtained from International Journal of Systemic and Evolutionary Microbiology, and also I have an analytical profile index test that constitute physiological data for identification of strains. So, well, and then we have Phytopath, and the Phytopath was maintained by Rothamsted, but unfortunately it is not maintained now. It's no longer available. So some of them are losing their importance. In the sense, funding is not available, they have been given up. So you need to select the database which will be with the available for a longer period. So maybe they are available, but they are not, new ones are not available. This is an archive website and no longer maintained. So they may give up. So we need to know about that. So we have a pathogen host interaction. Yeah, Fibas is again a database about the interaction, not only just the microorganism or pathogens, but also about the different hosts. So there are some interesting papers which are available in nucleic acid. I mean, I'm going to just show you, like, so that you understand. What do you really know in the sense? Now you need to know like what are the new technologies or platforms are available for sequencing, what kind of developments which are going. Read about them. And I find like this paper was very interesting. DNA sequencing at 40, past, present, and future. That will give you an idea about what more you can expect in the future. So that is available in, you can note down from this one. And this is the original paper of Sankar. You also read it. That's an interesting piece of information. I mean, like for which he got a Nobel Prize, and then DNA sequencing milestone, which are all given in the paper, like how do we go in the future. And every year, I think, every year nucleic acid comes out with the databases. Volume 51 database issue, January 2023. So January month, you go to the website of nucleic acid and read about the new databases which appear. And this every year beginning, you have new volumes, and if you go through all of them, you will understand about several databases which have been published in the past also, right? So they write about the acid database issue, online molecular biology database collection. Every information is available. So, and this is about EMBL at 2022. This is the same issue. Database resources for the National Center for Biotechnology Information 2023. It's again in the same issue. And I found database resources of the National Genomics Data Center, China National Center for Bioinformation in 2023. Right? So, and there is also a society which is known as International Society for Biocuration, and they have a paper database. So if you are developing a new database, you can also publish in this journal, right? Well, this is my last slide only to show you, like as your circle of knowledge grows, the diameter of ignorance increases. Okay, so more we know about our organism, we also understand we don't know many things about the organism. So does the circumference that is in contact with all the darkness outside the circle of light. So even if you try to learn, so ignorance is going to be there. So I'm only showing you from sequencing to the databases. Appreciate both so that, well, you have more knowledge. Well, I don't want to complete the other sentence, right? So for you, I'll say, well, these databases are important. Don't ignore it, because every single rupee or money you spend on your sequencing work or interpretation of your data, if it is not done with the right databases, your interpretation or your story or your understanding about the organism is going to be inadequate. Okay. Okay, thank you very much. Any questions? It's good. 5 minutes only. Why it is? Thank you for your attention. Attention is missing, right? Anything you want to learn? Cultures, yeah. I think it's a little overwhelming. Okay. No, initially when we thought sequencing is going to give us a lot of understanding, but now we know we have too many sequences. What to do with the sequences, right? So same way, like whatever you spend a lot of money. Not only you, every one of us are spending money thinking that we will get new information, we will sequence, we will get new information. But information is already available, I'll say, in the dark matter. All you have to do is, you need to understand, you need to, use your gray matter to understand your dark matter of sequences. Unless you get your understanding correct, the information is going to be unused, not available in a way it is desired. So you need to know like what are the tools to be used to bring a better understanding of the information which is available with you in terms of sequences. Well, so don't miss the databases. Spend some time, at least 30 to 40% of your, your research work should be always to keep track of all the databases which are coming up, so that you have more time for your analysis than spending time on understanding about like how to analyze them, because these are now databases will make things easy for you to analyze, right?
A couple of more points. Why this lecture is very important for all of you. First of all, you should know where the information is available to get into that information which you may need for your work. Number one. Number two, where you can showcase your data. It's a both way. You contribute so that some other researchers can really get into the data, can use it. At the same time, you also need somebody's data. Somebody was asking yesterday for designing primer. You need the genes, how to locate the genes which is used in qPCR as a reference gene. So you need information, and these informations are available in the public databases. So if you ask me who is contributing towards the databases, it is all of us, correct? All of us are contributing, and all of us are using. We are also a contributor and also a user. So that is the beauty of all these databases. Okay, having said that, two important points. All of us are handling biological material. It may be a plant material or microbial material or microbiome. And you are doing a downstream analysis to address certain biological questions. Correct? All of us are doing that one. You have some biological questions to be addressed, and you are utilizing the tools. What has been taught by the Netherlands team, what we are teaching here. For what? We are trying to make some interpretations. Those interpretations are going to add some knowledge in us so that we are better educated, better informed. 2 points you have to remember always. If you are handling any biological material, ensure that that biological material is preserved somewhere. Very often, you don't do that. We keep it in our lab, and there is a saying, the culture is in your lab is a riskiest thing somebody can do. Why? By the time you complete your graduation or your supervisor retires, the culture also retires. If that is happening, that is one of the worst things to have happened for a biological material. Why? The reason is the information you generated from the biological material that you have already uploaded in some database. It is already accession. The number has been already assigned and you have been using proudly in your literature and your thesis. But the corresponding, the source material, the biological material, if you ask, very often we miserably fail. And we come out with an answer that, sir, I kept in my lab, the incubator did not work properly, there is a power outage, my supervisor went on a transfer, all kind of excuses comes. To solve all these problems, if you go back into the genomic era when it was really taking inroads in science, a couple of very good white papers appeared in literature. The do's and don'ts for genome sequencing. That includes, of course, the metagenome sequencing, what you have been taught so far. The first point, what they say is, you try to preserve your biological material in some public database, public repositories. You accession it. This accessioning is an integral part of your thesis project or research project. Remember, nobody is going to force you to do that. right? That enthusiasm to do that should come from within. You have to come forward to deposit your culture, some culture repository. In our country, we have an excellent repositories in many places. We have repositories in Pune, in Chandigarh, in Mau, of course in Delhi also. Somewhere you accession it, and you may have to pay some money also. Of course you have to pay money because the culture is being maintained by somebody. What is the meaning of the word maintained? It is not putting the culture in the rack and you go for sleeping. No. You have to take care of the culture. What do you mean by taking care of the culture? You have to maintain two things. One is the viability of the culture, and second thing is the stability of the culture. What is viability and what is stability? Viability is to retain its life forever, number one. What is stability? If it is a pathogen, it needs to be pathogen. And one fine morning, it should not cause a disease means then it is gone. The stability is gone. So for that purpose, you have to pay money. So remember, whoever is involved in any genome downstream genome sequencing work or metagenome work, ensure that the culture is deposited somewhere with some accession number, so that in future, if somebody is found interest in your data, and he may look forward to the original culture what you handle. And if you are not supplying that original culture to the person who is in need of that, whatever effort you did till that point is of no use. Maybe it might have fetched your thesis degree, but it is of no use for anybody. You got my point? From that perspective, the lecture what Ramakrishnan delivered today is very, very important, and that gives you different options and where you can deposit the culture, where you can access information about the culture and the sequence data, where you can deposit your sequence information. These are all very crucial for our future science as well as the science what you are going to contribute for the next generation. And don't ignore that. Okay. Now come to the final part of the metagenome preservation. I put a question to all of you. Of course, you are isolating a single culture using different techniques, what we told yesterday, quadrant state, high tip method, purest to form. Okay? Divide of all other neighbors and all other environmental factors, you put it in the comfort zone, the culture is preserved somewhere. Okay? Come to the question of microbiome. Can we preserve microbiome? If so, how? Okay? It's an open question. We will come back to the same question again in the next presentation or next presentation. I want some answers from all of you. If we can preserve microbiome, whether it is a rhizosphere microbiome or phyllosphere microbiome or endophytic microbiome or whatever may be the microbiome, can we preserve it? If so, what should be the strategy for preserving the microbiome? Okay. Number 2 point, as of now, we don't have any repositories for preserving microbiome, but can we propose something? A microbiome repository for a country where we preserve the microbiome of specific plant-associated niche. Somebody is extracting a microbiome from phyllosphere, somebody is isolating from rhizosphere. From that particular sample, from that particular time point, can we preserve it somewhere so that you found some interesting data? Roy Maker was talking about some new cultures they found, they are trying to isolate that. Right, you may not have time today, but tomorrow some other student joins and you can go back to the microbiome and ask him to search that particular interesting flora in that particular microbiome sample. It gives you a lot of opportunities, right? So the question here is, can we preserve microbiome? If there is a possibility, come out with your original ideas, how it can be done in future. Okay? With that, thank you so much, sir, for making this presentation. It's a voluminous information he brought here, and excellent presentation. And I was really seeing the slides from the backside, quite a lot of information. Thank you so much, and please get into that presentation. It is already there in YouTube, and try to understand the fine details of those culture collection and the genomic databases. Thank you. So we'll break now. We have a tea break. The next presenter is already waiting.
Okay.
And 15 minutes. Come back by 11:30. Thank you.
Okay, this is not as a speaker. I just wanted to share an information with all of you. As a microbiology student, in the beginning, I'll say beginning of my career, I never thought of endangered species in microbiology. Okay, I never thought of, I would never think of any organism is going to become an endangered species. But when I open WFCC website, there is a questionnaire now on endangered microorganisms. It's a bit shocking for me because that is the way like we are now thinking about, because also there is some relationship with the endangered plant species, endangered animal species. When we lose an endangered plant species, an organism which is lost, an animal or a plant system, we are going to lose several endophytes once for all. And those microorganisms which has got a very close evolutionary relationship with those endangered species is lost, right? In addition to that, when I was writing papers in the beginning, reviewers used to ask me, can you prove that this microorganism is not available in this environment? It's very difficult to even show that this organism is not available in a particular environment, because we go by everything is everywhere. It is an environment which selects. So that principle was applied in microbiology. But now I could see that there is a questionnaire which is available in WFCC for endangered species. So, well, we are the preservation which he said, not only in terms of the cultures in the laboratories, but also in the nature is very, very important. So if we abuse the microorganism in the nature, we are going to be in trouble. Thank you. Thank you very much. So it's more.
going to help us mostly for our day-to-day genomic research, validation part. That is all about nucleic acid estimation and quantitation. We have been using PCRs, real-time PCRs. Yesterday we talked in length about that. Here another technology has come, right? So 2 days back we witnessed a lecture on PCR and qPCR. Variants of PCR we talked about. Now we have a new technology what is called as a ddPCR, digital droplet PCR. And Bio-Rad is one of the pioneer in developing the technology. And you please listen to the person, the expertise, the person who is having the expertise in this particular field. I think it's better to know from them directly what is the RT-PCR or ddPCR. I request Dr. Ramakrishnan to engage this.
So is it working? So this, yeah. This is if you, when your discussion is there, if you want to come here, you press this one. It goes off. If you press it again, it will come. This is next slide. This is previous slide. This is right.
And I like to share my experience with the PCR. Right. When I was a student, one of the favorite questions by many examiners is what is PCR? So all of us invariably in Viva, qualifying examination, they will ask about PCR. Right. So when I happened to be in Germany, I saw real-time PCR. So that was the time they introduced real-time PCR. So I was trying to buy it, and it took almost 10 years for me to get one system in India. But now, then later on, after I got my real-time PCR, I could understand the power of digital PCR. So
in front of all of us and on this stage actually. So as the topic says, accelerate your high quality research. Okay, so means whatever you are doing with PCR or real-time PCR, this ddPCR definitely is going to add value actually to the work whatever you are doing by those techniques actually. So we will discuss in further slides how it adds value and how it is better than other existing techniques. So like if you see the PCR history, okay, we all know normal PCR, conventional PCR, if you have seen Bio-Rad PCR in this lab, T100s are there, C1000s are there, S1000s are there. So those are actually conventional PCRs where you have to prepare your reaction, you have to run like it for 1.5 hours or 2 hours, and then you have to run it on a gel. Okay, so that PCR we consider as first generation PCR because with that only when Bio-Rad came in market after acquiring MJ and they introduced multiple PCR models in market. So this was the first phase of commercializing PCRs in market. Few other vendors also came, but Bio-Rad came as MG. So people who have used PCR since long time, they must have seen MJ research instrument in market actually or in labs. Okay, so that we consider as normal PCR, and nowadays also it is continued for many applications. So we cannot say that ddPCR is going to replace all the other techniques, but it adds value. So that suppose you have some applications which are with which you are facing challenges actually, in those applications ddPCR can help you in much better way actually. Okay, real-time PCR. So real-time PCR is also like a advanced form of PCR where you are not running a reaction on a gel actually. Whatever is going on in the tube itself, you can see the result during run itself. Okay, so you are making a reaction and very rapidly you are able to see the results. Okay, so this is real time. Real time means in live mode you can see the results. So it was very helpful during pandemic season actually in last 2-3 years, and a lot of labs who were not even doing PCR but they like very quickly they shifted to real-time PCR directly, just because it was the time of like it was the demand of that actually period. Okay, so here you prepare a reaction and within like 1 hour or 45 minutes you are able to see the results and you can report something that it is positive or negative, or you can do quantification. Okay, so now like the third generation we consider is digital PCR. So digital PCR is not a new technique, it is in market since long time, but it was in different forms. But we can say that without support from different industries or different manufacturers, any technology cannot be commercialized. Okay, it is in market or it is in labs, actually it is existing, people are doing, but even then, like if someone is not making it in an instrument which can be commercialized, which can be supplied to labs, everyone is not going to use that. So Bio-Rad is the first one who came in market actually, and they introduced this technology. So before Bio-Rad itself, actually Quanta Life actually there. So who prepared this technology and they commercialized, and Bio-Rad immediately acquired them, and then they commercialized it for whole world actually. So this we will discuss more about this third generation PCR, digital PCR. So Bio-Rad named it as droplet digital PCR. So name itself tells that we are doing PCR in droplets actually. So this is a little more detail, that after doing a PCR we have to run a gel, and on a gel you have to see the bands through ethidium bromide or Savasif staining, and by seeing these bands actually you cannot quantify them correctly. Okay, you can just say that whether it is positive or negative, and suppose if the concentration is very low, in that case sometimes bands are also very faint actually. So you cannot quantify or you cannot detect the targets which are very low abundant or which are not expressing in enough concentration. Okay, so here lot of limitations are there, and to overcome those limitations you can use real-time PCR. Okay, in real-time PCR you can do quantification, you can check the results in live mode. Okay, but here are also multiple limitations again. So suppose you want to do absolute quantification. So in absolute quantification you need to compare it with some standard curve. So every time in your experiment you have to do like a serial dilution series of a known target, and just with that you have to prepare a standard curve, and with that only you can do quantification. Okay, so every time you have to run standards. So I hope you all are aware about real-time PCR perfectly, so I am not going in detail in that. Okay, if you have any doubts you can interrupt me anytime. Okay, if you have any question, or like I don't want that suppose you have any doubts and just because of that doubt you are not understanding further presentation, so you can immediately resolve that. Okay, so now come to ddPCR actually, how we perform ddPCR. So in ddPCR actually we are doing partitioning. So how partitioning works? So we prepare a reaction in a tube in a 20 microliter or 25 microliter format. Okay, and then we divide it into multiple droplets, then amplify the target actually in the droplets itself, and then we read it through a reader actually, and just after reading result displays on the computer, and by that actually you can say whether sample is positive or negative or what is the concentration. Okay, so if you how it adds value, that I am just going to tell you. Suppose if you provide this smoothie to someone, okay, and just offer this and just ask him that how much fruits were used actually to prepare this smoothie. So no one is there actually who can tell that all the fruits whatever were used actually, he cannot name all. Okay, suppose some were used in very low concentration or low amount and some were used in very high, so the other fruit will overcome, and it will hide the taste of other which were used in low concentration. So if you give this platter, fruit platter to someone, then easily, like even without tasting, someone can tell that which fruits are there. Okay, similarly, suppose from a pond, and out of that suppose you want to count the number of a particular variety. So in a pond, it is very difficult to identify that or count that, okay. But suppose if you partition it into small chambers, small aquariums or small jars, easily you can count it and you can tell that how much number of different species are there, okay, accurately you can do that. So on the same principle actually this technology, okay, and here whatever partitioning we are doing, that is random partitioning, we are not limiting it by any means actually. So how it works? Suppose you prepare a reaction in a bulk mode, like you have prepared a 25 microliter reaction. So in that, suppose if targets are available in very low abundance, it is difficult to detect them. Suppose even you are using real-time PCR, but in real-time PCR also, there are limitations. Just because of that, you cannot detect very low abundant targets. Okay. Suppose the signal is very low, or suppose background fluorescence is too much, or suppose lot of inhibition is happening. So multiple factors which can affect real-time PCR also. Okay. And suppose you are doing PCR only, then if you run on a gel, if the target is already low abundant, then you will not see any band on the gel actually. But when you divide it into small droplet and you do the reaction in droplets and you check each droplet. So when you are checking each droplet means you are screening every partition. So if any amplification happened in any partition, easily can be detected. Okay. So how it it is like adding value means input target counting easy. It is like very easily you can do whatever DNA or whatever RNA you are adding actually in the reaction. You can easily count the copy numbers. So copy numbers means how many copies of that particular target are available in that reaction. No relative quantitation means in a reaction, whatever copies are available, it can tell you independently. Independent means if you run a single reaction in ddPCR, with that also you can tell how much copies of that particular target are there. So it is totally independent. There is no requirement of any standard curve, okay, just to quantify that. So how it converts that actually, that we will discuss further. Endpoint measurement means we are not doing any in real time mode or in starting, like after completion of full run actually it is being counted at the end. So means whatever is amplified that will be calculated actually. High precision means reproducibility and discriminability. Like suppose you do same experiment multiple times, even then you are going to get same data. Or suppose like multiple students are doing same experiment on different days, even then you are going to get same data. So it is very much reproducible and very easily it can discriminate different targets actually. Suppose you are doing a mutation detection, so in high concentration of wild type population it can easily differentiate that. Okay, so for that you have to just use probe chemistry. And then high sensitivity means in a rare population you can detect a bigger background, you can detect a rare population. Means suppose you have taken some soil sample and from that soil you just want to identify a particular species of bacteria. So you have to just design a primer and probe for that particular bacteria, and if it is amplifying can be detected here. Okay, which is like limiting factor in multiple other techniques. So just let us understand the Bired history actually in this technique. So initially, like in 2012, Bired launched QX100 actually. So QX100 was like only compatible to probe chemistry, not compatible to evergreen chemistry. So in normal real-time PCR, you can do probe chemistry and evergreen chemistry, but here it was not compatible to evergreen chemistry. It was only for probe chemistry. Later, they introduced QX200, which was compatible to evergreen and probe, but manual droplet generation. After that, fully automated. So suppose you don't want to do any manual pipetting, you just prepare your reaction, place your reaction in auto DC, and then it can convert all the reactions to droplet easily. Then came actually like QX200 IVD model. So like it is for diagnostic labs who just want IVD. Then auto DC with IVD. Then again launching of BCR AVL kit. So this is related to some like MRD, like minimum residual disease evaluation. Then another apparatus came, QX1. So this is different model than all other previous. So QX1 is 4-channel based and totally like in a single box. So here there are multiple different parts, like one PCR machine, one plate sealer, one droplet generator, and one reader, all are inside this only. So everything inside a single box. So this is very much suitable for high throughput, like you can run 5 plates together, like in a back to back mode you can run 5 plates. And then recently Biorad launched QX600. So like whatever instruments QX200 or QX100 were there, those were 2-channel based. QX1 is 4-channel based, and then QX600 just on the basis of demands from different labs actually, Biorad introduced QX600. So this is 6-channel based. So you can do 6 target multiplexing straight, or otherwise by changing the concentration of different probes, you can go up to 12 target multiplexing easily. Okay, so and continuously this product line is improving, so like lot of focus is there in the market, and in future this technique will be utilized very exhaustively actually. And in India itself, there are more than 85 installations of this DDPCR QX200 itself. So like it is quite evolving in India. So these are different models. Just this is automated generator, and this is reader. So reader is main component for reading the droplet. Droplet generation can be done by this manual droplet generator or this auto DC. This PCR machine is common, but this is not exactly the same which you are using in your labs. It is deep well PCR machine, then a plate sealer, and these are cartridge holder, cartridge and other consumables, oil and other things which will be used during full workflow. Okay, so anyone can buy this combination, like auto DC with reader or manual DC with reader, and these two are fixed actually. So this is live example, like in a lab it was installed recently. So this is actual like auto DC, then reader, computer and other things, plate sealer, PCR. So maybe if you visit to some ICMR lab, you can notice like multiple installations are there in ICMR labs. Okay, so here like you have to just prepare your plate, just place this inside this machine and just select the lanes, that how many lanes you want to convert to droplets. Automatically it will convert to droplets, and then you have to just seal that plate, place it in PCR machine, and then after PCR just read it. So how whole workflow is happening, just to show that I am just showing one video easy, so that easily you can understand that how it works actually. So meanwhile, if you have any doubts, please ask actually.
Can anyone tell how the droplet, digital droplet PCR is different from other PCR applications? No, I'm not talking about the ability. How exactly the amplification is carried? Where exactly? Not how exactly. Exactly. The amplification is happening in a droplet. So what is the droplet size? Nobody is asking that question. And
Yes.
how many DNA molecules are there in the droplet? These are all the basic questions you are supposed to ask if you want to really learn about digital droplet. Okay, what should be the DNA molecule inside the droplet? How many molecules? At least one, right? If you answer that, at least one should be there. Correct? So we expect one molecule in every droplet. Anyway, listen and then you will learn more. The PCR reaction is not carried out in the field as it is. Laboratory is carried out in the droplet. Where the template is.
Okay, so I'm just playing one video just to show you full workflow in short. So suppose you prepared a reaction or whatever samples you want to run, you prepared a reaction in a 96 well plate. So in place of 96 well plate, you can prepare in simple PCR tubes also. So here we have prepared in a plate, so that's why we are using multi-channel just to make it easy. Okay, so just aspirate your reaction, 20 microliter reaction through multi-channel or single channel, and put that reaction in the cartridge actually. So this is cartridge. So in this cartridge you can see there are 3 different rows actually. Okay, so in these 3 different rows, the middle row is for sample, the lower row is the like left side row is for oil, and the last row is for generated droplets. So first of all you have to put your sample, then by using multi-channel or single channel you add oil. So here whatever sample we added, that was 20 microliter reaction, and oil we are using 70 microliter. Okay, so here now after adding sample just add oil. So this is droplet generation oil. Okay, so there are 2 types oils, like if you are doing evergreen chemistry then you have to use evergreen oil, if you are doing probe chemistry then use probe oil. So the composition is different for both oils, but like by seeing or by just viscosity you cannot differentiate. Okay, so now after placing it inside this droplet generator, how it converts to droplet actually you can see. So you can see that the sample was dispensed in middle row and oil was here, and these are the droplets, these are the bells for droplets. So whatever droplets will be generated, those will be collected in the last well. So all these channels, like in each cartridge, all these 3 wells are connected actually. So those are connected through microcapillaries. So these are microcapillaries. In this microcapillary, 1 is coming from sample well and 2 are coming from oil well. So oil is coming from 2 directions, sample is coming from 1 direction, and this sample is being coated by oil and breaking at this particular point. Okay, so when it is breaking, it is converting into droplets. So the bore size or the orifice is constant actually. So this way actually we ensure that most of the droplets are of similar size. Okay, so this way actually droplet generation happens. So it takes around 2 minutes just to generate droplet for 1 cartridge. Okay, suppose you are running only 8 samples, so it is just 2-minute job to convert, and then you can transfer it to PCR plate. So after converting, just use multi-channel or single channel, transfer the droplets from this cartridge to a same plate or a different plate. Okay, then seal the plate, place it inside the PCR machine, just do the thermal protocol. Okay, so when you are doing PCR at that time, whatever like reaction is there, so all the components were used, those are inside the droplets. So like you used DNTP stack and other components which are required for building the DNA, and then DNA tag polymerase, then DNA, primers and probes, everything you mix in 20 microliter, ensuring that every droplet should contain primer, probe and all other components. So the concentration of the all the components is quite higher here actually in ddPCR supermixes. So that way actually we are just making sure that every droplet should contain that material. That should not be a limiting factor actually. Okay. So this way, after doing PCR, again you have to just take out the plate, place it inside the droplet reader, and then start reading. So with a computer you have to just control this machine. So this is similar to a flow cytometer machine, where this needle is going inside the valve, aspirating full volume. So like whatever generated, that was 40 microliter. So we added 20 microliter sample, 70 microliter oil, but actually the generated volume is 40 microliter only. So in this 40 microliter, around 22, 23 is droplets and remaining is extra oil, just to like giving some viscosity or some suspension to this, so that droplets are not sticking to each other. Okay, so this aspirates, then singulates all the droplets, and then passes through it like detector. So it is like taking help from the oil. So in the system also there is one reader oil is there. Okay, so with the help of that reader oil, it is singulating the droplets and then plotting the fluorescence here in the software actually. So this is amplitude chart actually for suppose FAM, and this is for big. So whatever droplets in which some target has been amplified, those droplets are showing little higher signal. Okay, and the droplets in which there is no amplification, those are showing lower signal. So this is just amplitude chart. So higher are coming at the higher side and lower are coming at lower side. So this is for FAM only. This is for hex or big. So suppose you are doing a probe based chemistry experiment in which you have used 2 different probes, and both probes are for different targets. suppose one is for wild type, the other one is for mutant, or suppose one is reference and other is your desired gene. Okay, so both you can run together and the data will be displayed like this. So if any positive droplet is coming, that will come at higher side, negatives are coming at lower side, and you can verify whether all the droplets are like coming at a proper scale, or the droplets which are at coming at lower side, those are negative. Just to verify that, you run every time NTC, no template controls. Okay, so in no template controls, you have not added any DNA. It means the fluorescence for NTCs should come at the similar amplitude, okay, or similar height. So with that, actually, by comparing with NTC, you can set a threshold. So when you are setting a threshold, this red line, the software can decide that above this line everything is positive, below this line everything is negative. Okay, so it is just counting the droplets, and then it is considering each positive droplet and negative droplet, and for counting, then applying Poisson correction. So this whole calculation is based on Poisson corrections. Okay, so that we will discuss further in presentation. So after that, it gives some number. So suppose if calculation has been done, after that it shows some number in the table actually. So here one table comes, and in that table you can see that how many copies are for FAM target, how many copies are for big target. So just after reading, it is displaying the number. Okay, so it is not comparing the standard, so that's why it is giving you absolute data. So it is totally independent reaction. So in this workflow, do you have any doubts? Yes. So suppose you are doing probe-based chemistry. So you have to design your probes which are very much specific to the desired target. Okay, but in evergreen chemistry, sometimes it happens that your primer is binding non-specifically to other site, and it is amplifying some other fragment also. Okay, so when other fragments are also being amplified, in that case you will see rain type clusters here. So with that rain, you can identify that either your primers are binding at non-specific sites. So you can see a different cluster also at lower amplitude, okay, just because of non-specific amplification. Or suppose your primer dimer is happening, primer dimer formation is happening. In that case also you will see some new cluster actually at lower amplitude. So with that, actually you can identify whether some non-specific amplification is happening or not. And you can also do gradient PCR. So in this ddPCR, actually whatever PCR machine we are using, that is gradient PCR. So you can just run a gradient. Just suppose like theoretically the temperature, annealing temperature is suppose 55. So you run a gradient from 50 to 60, so that you just verify that at which temperature your clusters are coming very clean actually. Okay, so in this way actually you can just specify, or like you can just decide that which temperature is optimum for your reaction. Okay, so whenever you are doing any PCR, always you do gradient actually, just to verify that your primer binding is proper, your probe binding is proper. So whatever results you are getting, those are accurate, and those are working with each concentration of DNA. Suppose sometimes you are adding more DNA in a reaction, in that case it is working. But suppose when you are adding very less DNA, in that case it is not working. So you have to decide your annealing temperature in that way that it should work with all ranges actually. Okay. So we do a gradient PCR, we do serial dilution series also, just to verify whether it is working at all dilutions. Okay, so these are optimization options which should be done actually. So we are preparing in a reaction 20,000 droplets. Okay, suppose we are converting 20 microliter into 20,000 droplets. So whatever concentration you use, it should be this much significant that you see some positive droplets. Okay, so yeah, just I am coming to that point only. So suppose you are using 20,000 droplets. So if you are adding too much DNA, that you are saturating all your droplets, in that case what will happen? You will see only one population. If you are adding too much DNA that each droplet contains one copy or two copy or more than two copy, in that case you will see only one cluster. So it is not possible for software to discriminate that what are positive, what are negative. So it will consider that either all are positive or all are negative. Okay, with NTC you will set a threshold, and after setting a threshold it will decide that it is saturated and it will not tell you accurate number. Okay, so in that case we have decided that the range should be within 10 to the power 5. Okay, 10 to the power 5 copies. So what you can use maximum 10 to the power 5 copies in a reaction. So lower if you are using, it's good, but you should not use more than 10 to the power 5 copies in a reaction. Okay, so suppose whenever you are trying an unknown sample or a sample in which you don't know that how much concentration of DNA is there, always initially if you do the quantitation of the DNA, with that actually you can use less amount of DNA than higher. So this way actually you can run 2-3 different concentration, and with that you can just check whether at which concentration it is working. Okay, otherwise we suggest that you do dilution series, and with that you just identify that which concentration is working if you don't have anything to quantify your DNA or RNA. So it is similar to PCR or RT-PCR, like whatever components you are using, those are same actually. Quality may be different, like DNTP tech and other components, and primer and probe, and water and other many things which we use in a PCR or real-time PCR reaction. Components are same. In real-time PCR you are using evergreen dye, or suppose if you are not working evergreen or evergreen base, then in that case you are using probes labeled with different fluorophores like FAM, Hanks, Wick, or different fluorophores are there. Okay, so same type of reaction we are preparing here also. So suppose you are doing probe-based chemistry or evergreen-based chemistry, in your real-time PCR, you can easily migrate your experiments from real-time PCR to ddPCR. It is very easy actually. So Bio-Rad has given one tech note also. Just with the help of that tech note actually, you can just follow the conditions. So easily you can replicate your experiments in place of real-time PCR. You can do it in real-time PCR. Hmm. So those advantages I will discuss here. Okay, actually.
Actually, I had one small
Yes, sir.
You told 20 microliters is divided into 20,000 drops.
Yes, sir.
That means each droplet is around 1 nanoliter.
1 nanoliter around.
So, in 1 nanoliter, theoretically, how much of DNA can be accomplished?
So it depends on the size of the DNA actually. Okay, suppose you are using human genomic DNA. So in that case, each droplet can hold up to 4 or 5 copies, 4 or 5 molecules it can hold. Okay, suppose you are doing using plasmid DNA, then in that case size is very small. So more copies can be accommodated in each droplet. So when you are suppose you are working with a virus sample, so in that case the particle is very small, so more number of copies can be entered in a droplet. So whenever it depends on the genome size also. Okay, because droplet size is fixed. So whatever area is there in that, as many copies can be accommodated, that will enter actually. And this is random distribution. So if you are adding more copies, no control. If you are adding more copies, then more copies will be there in the droplets also. If you are adding less, then chances are there that each droplet will have one or two copies maximum.
That only means that certain droplets may not have DNA also.
Yes, it can happen. So suppose when you are adding less amount of DNA, in that case you are adding less copies. So when you are having it is random distribution. So if you have more number of partitions and less number of copies, so the chances are there that each copy will be segregated in different droplets. There are less chances that same droplet will have more number of copies actually, because during partitioning it is like segregating.
You try to evenly distribute.
Yeah, evenly distributing. Okay, so you understood all this. Like, if suppose we are using automated system, you can convert your reaction. You have to just decide how many lanes you want to convert. Okay, so this way actually you can use this whole workflow. So this is PCR machine, this is reader. Okay, here you have to place the plate and then read it. Okay, so this is very much similar to a flow cytometer kind of instrument. Okay, so this is workflow in like brief. So now whatever I explained actually, it is just a representation of that. You are preparing reaction. So for preparing a reaction, you are adding DNA, supermix, then other components, then partitioning, making droplets, then doing PCR, then reading this. So suppose any positive droplet is there, it is giving positive signal for this, suppose green, this is blue, so giving positive for blue, and then it is being displayed in this chart actually. So you can see in a sample, if total droplet number is like positive is high, then you will see darker. If the number is low, then it is like very lighter. So this way actually. So how much time it takes actually when you do your reaction? So suppose if you are running only 8 samples, then the droplet generation time is very low, like only 2 minutes. So in that case, you can finish this whole experiment in just 2 hours or 2 hours and 15 or 20 minutes. Okay, suppose if you are running more number of samples, in that case, like suppose you are running full 96 plate, so this droplet generation time will be more. In that case, your time will be added. So this droplet generation time added to the workflow, and this reading time also added to the workflow. Just because of that, it became 5 hours, around 5 hours. Okay, and even if you are using a automated, in that case also it is around 5 hours for reading a full 96 well plate. Okay, this already I explained, like sample you have to add in middle and then oil and other things. Okay, how it works, this part we have cleared. So now here you can see after generation of the droplet, you will see 2 layers actually in each well. So upper cloudy layer is for droplets and the lower layer is for oil. Okay, so if by verifying physically also you can check that droplet generation has happened properly or not. So suppose in any well, if you didn't get proper number of or like proper amount of droplet, you can just regenerate droplet for that particular well in further lanes actually. So you have to just add reaction in further lanes and redo the experiment, so that like after completing PCR or reading, you are able to get the data for that well also. So here actually multiple checking points are also there. It is not totally blind. So after making reaction, when droplet generation happening, you are able to see that droplet generation happened properly. Then after PCR also, you are able to see in the plate that your droplets are stable actually. So if it is not stable, then you can but before reading itself, you can just calculate that, or you can just assume that your results will not be proper in that case. Okay, or suppose some bad results are coming in a well, you can just identify that why all the bad results came for that particular well. Okay, so all this we have explained. So now this threshold part I explained, like you have to just set a threshold in between. So like you have to just compare it with a negative well or no template control well, and with the help of that you can just differentiate what are positive, what are negatives. Okay, so suppose if you are having any non-specific amplification, you will see a lot of rain here, or suppose you will see cluster at lower amplitude, or suppose any primer dimer happening. In that case, also you will see one more cluster here. So easily it can be differentiated. So now how to do multiplexing in this? So suppose you can do multiplexing straight away by using 2 different probes. One is labeled with FAM, other is HEX or BIC. Or suppose you want to do more target multiplexing. In that case, you can manipulate the concentration of other probes. So suppose for 2 target it is fixed. For 3rd target or 4th target, you are like reducing the concentration of the probe or reducing the concentration of primer. In that way, you are just changing the location of the cluster. So whenever you are doing a 2 target multiplexing, you will see 4 clusters in the form of in the 2D chart actually. So here data is being displayed in 1D chart. 1D chart is like only amplitude chart is there. In 2D actually different locations. So different quadrant it is showing for different clusters. So suppose all the negative droplets for each well are coming in at a single location. Okay, then in the droplets which have only FAM target amplified, those will come as a single cluster. Droplets which have only hex target or bit target amplified will come as a different cluster. And then some droplets will have both copies, like both amplified, FAM also amplified and hex also amplified. So those will come as a double positives. Okay, so here you can see 4 clusters are coming quite apart from each other. So there is lot of space in between, here, 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 here. So suppose if you want to do more than 2 target multiplexing, in that case you have to just adjust the concentration of your probe or primer in a way that your cluster should come either here or anywhere in between. Okay, so that part you have to do, and how to do that part, actually for that also Bio-Rad has given some tech notes. Just by following those tech notes actually you can easily do 3 target or 4 target multiplexing in a 2 channel based instrument, because here this is 2 channel based, so you have to use only FAM or hex labeled or FAM or bit labeled probes, but even then you can go up to 4 or 5 target multiplexing. So I will show some examples in further slides. So here how all the data patterns looks actually. So this is 1D plot, this is 2D plot for a well, then this is tabulated data. Here it is a like series concentration plot where one target is constant, the other target concentration is changing. Okay, so that also you can see in the software itself. So lot of information it provides. So now it is calculating the copy number accurately, but how it is doing that actually? So there are 4 examples of 4 different concentrations. Okay, so it is totally based on Poisson correction. So suppose in this negative sample you will not see any amplified droplet. So there are 143 droplets in each circle. Okay, we are making 20,000, around 20,000, but here I am just taking example of 143. Okay, so out of 143 there is no positive, here few positives because it is low concentration, then medium, then high concentration. So when you are adding more amount of DNA you will see more number of positive droplets. Okay, now if by droplet number only if you are calculating, you can easily tell that here it is 0 negative, here 6 positives, and here all negatives, here 6 positives, here 34 droplets are positive, so 34 copies at least are there. Here at least 70 copies are there because 70 droplets are positive. Okay, now when you apply correction, Poisson correction, it tells you the correct number. So here in place of 6 there are 6.2 copies, there are 38 copies, there are 96 copies, and it is taking like calculation, like it is considering so many parameters just to calculate this number, that if you go in detail in the software manual you will realize that it is considering that some droplets containing 1 droplet, some are containing more than 1 or more than 2 or more than 3 also. So it is considering all those things, then only it is giving you this number. Okay, so and if you compare this technique or compare the results of generated by this technique with any other technique, it gives you very reproducible data in comparison to any other technique. So suppose you are doing a dilution series, you do this dilution series repeatedly, you will see same number of copies every time. Okay, if your DNA is not degrading. So it is very much reproducible. So for that also, like multiple labs have done study with the same reference material. There is a publication also where one same material was provided to 21 different labs, and in all labs actually data was same. So the instrument was different, operators were different, but even then the final data was same. So it is very much reproducible. Now about multiplexing. So you can do multiplexing in evergreen chemistry also or in probe chemistry also. So suppose when you want to run 2 different targets in a single reaction, so you can do like multiplexing on the basis of primer concentration. So when suppose for one target you are taking less concentration of primer, and for other target you are taking more concentration of primer. So in that case, the concentration of primer is more, it means target will be amplified more. And here in each droplet the amplified target will be less. Okay, So just on the basis of that, amplitude will be different, and if you mix both in a single reaction, you will see this type of clusters. So this lower cluster is negative for all, this cluster is for this one, this cluster is coming here, and this upper cluster is super cluster. Super cluster means the cluster which contains both type of droplets, or the droplets which contain both type of copies. Okay. So with evergreen also you can do multiplexing easily. Similarly, on the base of size also, amplicon size, you can do multiplexing. For a smaller size, there will be low fluorescence, and for bigger size, fluorescence will be higher. If you mix them and run, then you can see 4 different clusters. So with Savarin or evergreen chemistry, it is quite possible with ddPCR. Okay, now comparison with real-time PCR. So in real-time PCR, suppose you are doing multiplexing, then you will see different amplification plots. Here you will see different clusters. Okay, so with these clusters you can identify whether this cluster is for wild type or mutant. Here similarly you can see the clusters are being converted into format of amplification plots. Okay, so when you are doing multiplexing for more number of targets, you will see this kind of pattern. So when you are running, suppose 4 target multiplexing, so you are changing the concentration of different probes. Okay, so here you can see 1.4 times concentration of FAM probe and 0.6 times concentration of FAM probe for other targets. So for one target we have increased the concentration, for other target we have decreased the concentration of probe. Both are labeled with FAM, but both will come at different location. Both are FAM labeled, but both are coming at different heights. Similarly, for other 2 targets, for hex also, for one target the concentration is less, and for other target concentration is more, and both are coming at 2 different locations just by changing the probe concentration. And so in first row you can see all the single plex reaction, or like droplets which have single target. In second row you will see droplets which have both targets, and in further row you will see the droplets in which 3 or 4 targets are available actually. So this classification can be done by software, and you can just nominate that which cluster is for which type of target, and accurately it will identify that what are the other clusters. So you have to just calculate for first lane, and for others it can tell you what is the concentration of target actually. So this way actually you can go up to 5 target multiplexing in a QX200 itself, which is 2 channel based. Okay, so here it is defining 3 target multiplexing. So these are negative droplets, these are single positives, these are double positives. So like for this and this positive are coming here, for this and this double positives are coming here, and for this and this, these are coming here in the middle, and this cluster is for the droplets which are having all 3 targets. Okay, because it is random distribution, so there is quite possibility that one droplet will have all 3 targets also. So 3 target multiplexing is very easy, and if you are increasing more than 3, then you need some optimization. Now how it adds value actually. So in real time PCR, if you are doing any reaction, okay, so it is like calculating your concentration in the starting itself. Okay, suppose here whatever amplification is happening, and which is just because of this amplification at a cycle the signal is crossing the threshold, that cycle we consider as a CT, okay, threshold cycle. So if this threshold cycle is shifting little bit just because of any factor, the whole quantitation will be like affected. Suppose in your standards are working fine, but in your sample reaction there are some inhibitors. Just because of those inhibitors, your CT value is shifting 2 number or 3 number. So even if one CT is shifting, means it is 2-fold change. Okay, or if suppose 3 CT is shifting, then there is around 10-fold change. Okay, so if CT value is shifting, then whatever quantification is happening just on the basis of a standard curve also, that is not accurate. Okay, but in ddPCR, even with a sample in which a lot of inhibitors are there, but if any amplification is happening, okay, if even poor amplification is happening, it can be differentiated from negatives. So if there is small gap between the clusters, like positive or negative, it can easily count that copy number. Okay, so it needs only small amplification. So like we can say that without like proper optimization also you can do ddPCR, or ddPCR is much more tolerant to inhibitors in comparison to real-time PCR or PCR. So this is the main advantage actually, because whatever inhibitory effect is there just in the biological sample, just because of anything which is being diminished just due to partitioning. So you are doing reaction in partitions, that's why it is diminishing the effect of that inhibitor, and just because of that you are getting proper results. So many times you run a same sample in real-time PCR and ddPCR, you will not get any data in real-time PCR, but you will see results in ddPCR. So suppose you are working with a soil sample or any plant sample where a lot of phenolic compounds are there. In that case also, if some amplification is happening, easily it can be differentiated. Here, suppose there is very small change in CT, okay, in two samples, that can be just because of like artifacts also, suppose, or some inhibition also, and it is very difficult to calculate that change actually in real-time PCR. But in ddPCR, it gives you accurate number. So in the number format, you can easily differentiate that how much fold it is showing. Okay, so suppose if there is two-fold change, there will be only one CT change here, but here it is in the form of numbers, copy numbers actually. So in this sample has 40 copies, this sample has 80 copies, means this is clearly two-fold change. So the results are coming in very like in easy format in the ddPCR, or independently also. We are not comparing it with any other thing. Okay, so these are basic advantages, like no need of a standard curve. So for any quantification, directly you can quantify the reaction. No worry of PCR inhibitors, means like if you compare with real-time PCR, whatever I explained in just previous slides, it is very much tolerant to inhibitors. No replicates are required because you are doing a single reaction in multiple droplets. So if you are doing reaction in multiple droplets, each droplet is working like a replicate. So there is no need to run more number of reactions. And LOD is LOQ, means whatever you can detect, you can quantify. Many times it happens in real-time PCR that you can detect, but suppose that is coming out of your standard coverage, then it becomes very difficult to quantify. But here you can detect and you can quantify also. So suppose in a reaction only 5 positive droplets are coming, even then it can tell you exact number of copies, that how many copies are there. Okay. So critical benefits we explained earlier also. Absolute quantification, it gives precision, very much precise. So you will see very minimal CV between replicates, or suppose if you are running same reaction in multiple times, very minimal CV will be there. Very much reproducible, very highly sensitive, very much accurate result it gives, and inhibition independent, means even inhibitors are there, it gives you some results. Okay, so this is example of different days experiment. Suppose same experiment was done, same dilution experiment was done on different days by different operators, even then the values are coming in the same range. And similarly, this example I was giving, same reference material was provided to 21 different labs. This is publication available, you can see. And out of all the 21 labs, only 2 labs were there where results were different, and remaining 19 labs were same. And in those 2 labs, the results were different just because of wrong thresholding, because they set the wrong threshold. Okay, and after correction of the threshold, in those 2 labs also results were same. So you can go through this publication later. Okay, now this is example of showing you again dilution series. So these are actual results from some experiment where we have done a dilution series in multiple replicates, keeping constant concentration of the background and diluting one target. And you can see in all series results are coming same, and if you see the CV, it is very tight actually. So for FAM target, for hex target, or for FAM so different operators have done this experiment, but even then there is very less CV difference. So now how it is able to detect very rare events, or you can say rare mutations or rare target detection. So in a bulk reaction, suppose any target is available in very less concentration. In that case, what is happening? Many other targets are available in high concentration, so the chances of detection of that target is very low, like 0.1% only. So suppose there are 1000 copies available for wild type or the background, and 1 copy is present for mutant or the desired target. Okay, so just because there are so much, it is not detectable. But when you do partitioning, so in partitioning, what is happening? Your 1000 copies are also being partitioned with this single molecule also. Okay, so this single molecule is also going in a droplet. Just assume that in a droplet with this 2 more wild type copies are also entering. Even then, there are chances of detection are increasing up to 33% if there are 2 wild type and 1 mutant. Suppose by chance if this single mutant copy is going into a single droplet, then also you are increasing the chances of detection up to like. So in a bulk reaction, it becomes very or to detect any rare target, but when you do partitioning, it becomes very easy. So you are increasing the chances of detection. So in that way, actually, you can easily detect any rare target in any type of sample. Now, rare gene detection, one more example. So when by doing dilution for one target, okay, so in that case you can see in real-time PCR at lower concentration it was almost same. So it became flat actually. So in all lower concentration wells it is coming almost same. So it is very difficult that all 4 wells are having same concentration, or it is just artifact, because here you are comparing it with some standard curve, and then only whatever number is coming on that you are trusting. Okay, but here in ddPCR it was able to detect up to single copy as well. So if it is able to detect up to lowest concentration, then it is very good technique for any experiment in which target of identification is available in very low abundance. So this is another example of like inhibition. Suppose you are running a real-time PCR reaction, and in real-time PCR reaction, if you run without optimization, so if you are running without optimization, it means whatever target amplification is happening, that is not appropriate. Suppose the CT should come 25, in place of that CT is coming 28 or 30 or 32. So the quantification will be totally wrong. Okay, so in that case, first of all, you have to do like proper gradient experiment, and then do proper dilution series for a standard. Then you identify that which concentration of DNA is optimum and which temperature is optimum. Okay, then only you start doing reaction. Similar experiment we did with ddPCR also. So this is example of same reaction in different wells at different temperatures. So this is 55 to 65. So when we are increasing temperature, so what is happening? Just because of high temperature, the binding of the primer is reducing and amplification is reducing. But even then, suppose in a droplet, in here at suppose 55 is the optimum temperature. So at 55, proper amplification is happening in each cycle. So means in this from 1 copy, 10 or 100 copies are being converted from 1. Okay, but in this reaction from 1 copy only 10 copies were converted, just because of this inhibition due to wrong temperature. Okay, so just because of wrong temperature, your reaction efficiency came down. That's why proper amplification didn't happen. Even then, it can be differentiated from negative. So if it is differentiable from negative, the quantification is same actually. So even without optimizing the annealing temperature, the final number is coming same. Okay, while in real time PCR it is not possible, even in normal PCR it is not possible. Okay, so here actually it is very much tolerant and very much accurate even at wrong annealing temperatures. Now another example of inhibition, here in this reaction you can see just because of inhibitory compound the total amplitude came down, but even then it is showing proper dilution number. Okay, this is by adding different inhibitors in a reaction. So here suppose in this reaction there is no inhibitor. So this experiment was done in QX1. In this 10% media was added, so that's why some clusters were skewed. Here HEC lysate, hexyl lysate introduced, here DMSO introduced. So even with inhibitors also, it is quite able to differentiate clusters, and if clusters are being differentiated means it is quite able to quantify things. So this is very important actually when you are quantifying copies in high concentration of any background or any unwanted product. Suppose you are producing any product in a cell line, and cell line DNA or cell line components are also coming in that product, even then you can quantify your desired target. So this is about cell and gene therapy. I am skipping this, okay, and this already explained, so I am not repeating, okay. Now something about assays. So with the instrument, Bio-Rad is also giving thousands of assays which are available on Bio-Rad website, and you can design your own assays also, or you can directly order the assays which are available on the website. So like multiple assays available which are already wet lab validated, or you can design your own assays and you can check whether those are suitable for ddPCR or not. Okay, so these are multiple assays available. So this already we discussed. This is whole cell workflow. These are some other slides actually for other workflow. So like this is application only. So for whole cell workflow, we can use this ddPCR also. So you can partition your intact cells with the help of this droplet generator, and then lyse those cells inside the droplet itself, then do PCR and then do reading. So like you can get data from a single cell. So for single cell workflow also this is being used, okay. Then in industries it is being used for contamination testing. So like Bio-Rad has multiple kits which are available for contaminant testing, okay, like mycoplasma contamination. So this kit can differentiate up to 100 different species by using ddPCR itself, okay. So now come to publications. So till now on Bio-Rad website you can see around 6300 publications, okay, which are available in the list. I will show you list here on the internet itself. So just you search this link actually, or you go on Bio-Rad website, just select digital PCR. In digital PCR, just at the bottom you will see on that page ddPCR publication link is there. Just click on that. Here all the publications, so just by reading abstract you can just identify whether it is of use for you or not. If you click on the link, it goes is available, and these publications are arranged according to different applications. So suppose you want to see any publication for crop improvement, or suppose for agriculture related, or for copy number variation, you can directly search on this. So these publications which are already reviewed by Bio-Rad, and their links are available on this website, okay. But if you go through internet only, like in Google, you will find more than 7,000 publications on internet actually. So every day some publication. So in all good journals it is available. So in like all the fields of molecular biology, you can apply it actually for agriculture, like for GMO testing, for environmental monitoring. Suppose you just want to see encroachment of a particular species, okay. And in NGS workflow also it can be used, for CRISPR workflow, for gene editing it can be used, for target DNA quantification it can be used in any sample, for gene expression or for single cell analysis it can be used, for pathogen detection. So like for animal pathogen, plant pathogen, for anything it can be detected. It can be also, it is very useful in like mutation detection, and very widely being used in cancer applications, okay, for liquid biopsies, like for urine, blood, or any body fluid, it for detection of mutations or for detection of copy number variations, it is being used. So in agriculture, I think most widely being used for CNV experiment, copy number variation experiment. Suppose if you are doing transfection of a gene in a plant, okay, and then you want to just check how many copies are, okay. So in that case you can do quantification with the use of real-time PCR also, okay. But there are certain challenges with real-time PCR. So suppose when the variation is more real-time PCR can easily differentiate, but when the variation is less, it is very difficult. Suppose in one sample, in the reference there are 2 copies, and in your target there are only 3 copies. So it is almost 50% difference. Okay. When in place of this, actually, if you take an example of, suppose in your reference there are 10 copies and in your target there are 11 copies. Okay. So there is only 10% variation. So if you quantify this in real-time PCR, it is very much difficult, because it can be just because of due to any artifact also, due to inhibition also, due to multiple regions. But in ddPCR, it can be counted reproducibly. So if time permits, I can show you those slides also. So these are multiple applications which we seen in previous slide. So like liquid biopsy, copy number variation, detection of rare sequences for viral load, for any pathogen detection in infectious diseases also. Okay. So these are few publications I have just taken, which are related to agriculture field. So you can see all these publications on internet or on Bio-Rad website. Okay. And few more for which are available for crop improvement, in which ddPCR has been used. Some are very recent also, like in 2020 or 21 also. So this is so like all these are available on website. There is no need to search on internet. Directly you can go on Bio-Rad website and you can find any publication of your interest where ddPCR has been utilized, so that you can understand that how for your application or for your work it can be used. So multiple publications are there. I have just taken some examples. So this we have already discussed, and this is cost calculation kind of thing, that how much it is like comparable to real-time PCR. So it is very much equal to real-time PCR. So suppose in real-time PCR, if you are running one sample, you have to run a sample into duplicate or triplicate first thing. Second, then you have to run standards, okay, just to generate a standard cup. So suppose for quantification of single sample also, you are running multiple reactions, okay. But here for quantification, you are running only single reaction. So suppose you are running only 2 reactions, one is NTC, one is your sample. Even then you can quantify easily. So it is very much comparable to real-time PCR. Even sometimes it is cheaper than real-time PCR in some cases. Okay. So it depends on your experiment, but it is not. But after that, your day-to-day handling is not that much costly. So with this, actually, I just want to end this presentation. I can show you multiple other slides as well, if sir permits. Or we can see Bio-Rad website. So this is Bio-Rad website. Okay, just go to life science research. You can see digital PCR. So just click on digital PCR. So here you will find all the topics related to ddPCR. So up to 1 kb you can amplify in ddPCR. Can you? Yes, you can take actually. So for sequencing you want amplicon, okay. So in that case you have to run duplicate reaction. One reaction you have to read just to verify that your experiment has worked properly, and second reaction without reading you can take it out and you can isolate the DNA. So there is a procedure, 30-minute procedure actually available in user manual itself. There you can see that how to take out the amplicon from the reaction. So it is very easy actually, easily you can do. Yeah, yeah. Yes, sir. No, sir. Around 1 nanoliter. So around 0.9 or 0.85 nanoliter. Yes, sir. That contains actually. So this only we are not saying actually. You can go on NMI website like National Measurement Institute. It is present in Australia. So they are continuously doing evaluation for the droplets being generated by Biorad, okay, in different instruments like in QX100, 200 or in other higher models, QX1 and QX600. So it is very much consistent also, and they have verified also that the components are available actually. Because if components are not there, then how amplification will happen? Yes.
So in one of your slides you have mentioned that because of the threshold changes the results will vary. What is the threshold?
So threshold means cutoff actually.
No sir, how can we set the threshold for this?
So suppose you are running one reaction and you are running one NTC, no template control. Okay, so in no template control, when you are not adding any DNA, in that case, all the droplets will be negative. So all should come at lower amplitude side. So we have to set a line actually that below this line, all the droplets are negative, so that in sample also it will consider that below this line, all the droplets are negative. Okay, so just to set a partition actually, that if suppose if in a sample you are having only droplets in a rain format. Okay, so in any sample, if suppose, let me show you. Here, okay. So in this actually you can see what happened. Or in this slide you can see, okay. So here you can see two clusters are there, okay. So these colors, two colors are there, one is blue, one is black, okay. So machine is not deciding that what is positive, what is negative, okay. We are deciding. So to decide this actually, we run one more well which is known as NTC, no template control, okay. So in real time PCR or in PCR also you always run one no template control, so that you are sure that whatever amplification is coming, that is not just because of any contamination. Suppose any contamination is happening, in that case what will happen? This amplification you will see in all wells. So if you are not running an NTC, then in that case you will consider all samples as positive. That is not true actually. So you to verify that whatever experiments you have done, okay, that is true amplification, the amplification is true. For that you need NTC first thing. Second is to define that below this line, below here everything is negative, above this line everything is positive. You need one more val in which you have to run NTC. So in NTC you will see only this population, only this, not this. Okay, it is actually it is not connected with my laptop. Otherwise I could have shown you that. Okay, I can show you separately in my computer. Okay, so when you are running NTC, so in NTC one more val is here in which only this population is coming at lower side. So you have to just, there is option in the software for threshold. So just click on the threshold and just click here. So there one line will be drawn here, and that line decides that below that everything is negative, above that everything is positive. Okay, so that threshold, for deciding that threshold, actually you need NTCs.
So, but what if our copy number is even lesser than that threshold value?
No, your copy number is lesser. In that case, you will see this cluster is very rare kind of thing. Like this is denser, means more number of positive droplets are there. If less number of positive droplets are there, you will see less number of positive droplets here. So less number of dots actually. So each dot is a droplet actually. So if suppose you have only 10 copies in your reaction, so you will see maximum 10 number of positive droplets in that case. So here the concentration is very high. That's why you are able to see this cluster is very dense actually, because more number of droplets are having. So suppose as I shown you that chart in which 4 samples were run. In one there is nothing. In second there are very low number of copies, and then medium concentration, then high concentration. So as much concentration you are increasing in this reaction, more number of positive droplets are increasing. Okay. So maybe just a minute I can show. Yes, sir. Yes. Yes, sir. No, sir. So, like this is dependent on threshold only. On the threshold actually. So suppose if you are setting a threshold, so above that everything it is considering positive, below that everything. Yeah, so you decide with the help of no template controls. Yes. So that's why only we are running NTC actually, because we have to decide that where we should have that line actually. Huh? Everything is positive. Yes. So, but in NTC you are not adding any DNA. So why X? Yes. Okay. But if there will be any basal level expression of gene, it will come above the NTC. No. So it doesn't happen actually. So just let me tell you, this marker is working. Any pan marker is there?
So, we can answer that.
Yeah.
I think EPCR you attended. So I told there will be a basal level expression. The machine cannot capture it.
Yes, sir, but this is very sensitive. We know that even if the signal
Yeah, but it is again I'm telling you, the amplification add up to the fluorescence intensity.
Yes.
Correct? The point at which the machine is able to see that, the CQCT value we discussed that. That is the actual threshold. Beyond that, the machine is able to see that. This is already discussed here. I am really surprised by the question what you are asking here.
sir, but actually in your application we are taking housekeeping.
I got your point. There is a G, but untreated control. That's what you are asking.
Untreated
Yeah.
housekeeping.
It's not an absolute control. Untreated control, there the template is there, but not the treatment. Correct. What he is asking, in the absence of any treatment, the template is there in the droplet. There would be a basal level expression, basal level fluorescence.
So just let me tell you. So suppose when you are adding DNA amplification for your target, okay, just because of that DNA also, the fluorescence of that total droplet will change. Okay. So you run two reactions. In one you are adding only water, in other you are adding DNA. Okay. So in the droplets in which only water is there, those droplets will be there at little lower amplitude in comparison to your sample. So, or suppose you run two reactions. In one reaction you have added only probe, in other reaction you are not adding any probe. Okay, you are adding only primers. Okay, so in that case, when you are not adding any molecule, just because of that fluorescence will be there, it can show zero droplet in the other well, because there is no fluorescence, so it is not detectable in the system. Okay.
Not detectable in the system. That is the most important point. To try to understand.
So
We can have some more discussion also.
yeah, I can show you here data. Actually, this is not connected to my laptop. Otherwise, I can show here in my laptop.
You got your question answered or not yet?
So in software itself, I can.
You need to explore a little deeper.
So whatever, so this software is just displaying the fluorescence coming from the droplet. Okay. So suppose just understand that suppose any other component is there, other compound is there, which is just creating some nonsense fluorescence in that droplet. So just because of that also, the reaction in which some DNA has been added, it will give you some basal extra fluorescence. So suppose in NTC it is coming as 100, in your sample it is coming at 150. But when there is any amplification is happening, the fluorescence difference will be more actually, in thousands actually. So
That is what I was asking. Like, why don't we keep the reference level as a baseline expression?
so you can in place of
Anyhow, it will be zero expression because there is no template.
yes. So in place.
No means of that.
So in place of adding barter to your no template control, you can use same media or same medium, suppose TE in the reaction itself for NTC. So you are keeping background same. So in that case, when you are keeping background same, your total basal fluorescence will be same. Only fluorescence just because of extra DNA will be little higher. So suppose you have added DNA in reaction, okay, and there is you have added high concentration of DNA in the reaction, and out of that high concentration, whole DNA is not being utilized for amplification. Okay, a lot of DNA is unused. Okay, just because of that unused DNA also, some added fluorescence will be there, but that will not be that much that it can differentiate or it can overcome your positive droplets. For your negative droplets, suppose your fluorescence is coming 100 or 200 or 1000, for positives it will be 4000, 5000 or 10,000 actually. So difference will be huge actually. So easily differentiable actually. So in that chart also, like it is not there, you can see that amplitude is coming in thousands.
The band will come.
Yeah, band will come at little higher side actually.
When you are discussing about complexity, sir has explained that changing the concentration of the primer will have different peaks. So can you elaborate on that?
So when you are changing concentration of primer, in that case what will happen? You are limiting reaction efficiency. So when you are limiting reaction efficiency, suppose in a droplet there is one copy of DNA. So with that one copy, if any component is not in limited concentration, what will happen? It will amplify up to the saturation level. Okay, so when saturation reached, at that time it will come same, but you are not letting like it to reach to saturation level. Okay, you are not allowing that reaction proceed with proper efficiency just by diminishing or just by reducing the primer concentration. So if less amount of primer is available, in that case what will happen? In first few reaction efficiency will be good, but after that when the material is in limited amount, in that case that product will not amplify with 100% efficiency, and suppose in first cycle there was one copy, so from 1 to 2, then 2 to 4, 4 to 8 or 8 to 16, it has happened. Now primer is not available. So what will happen? In next cycle from 16 to 32 conversion will not happen. It will come down actually. And then again in next cycle it will be less, and at a particular level it will be just for amplification was supposed to be done, that has been happened. But in other reaction you are not limiting that thing. So you are not limiting, so it is proceeding to saturation. So you have to limit in a way that it should come at different level. No. So because you are comparing with NTC only, then why different threshold will be there? Threshold will be same. So there will be 3 lines in a, suppose you are doing multiplexing just because of primer concentration. So in at the bottom there will be negative droplets, then at little higher side droplets for which you have added less amount of primer, then at higher amplitude droplets for which you have an optimum amount of primer. Yeah.
There could be changes in the amplification efficiency if you are limiting the primers by one multiplex.
Yes, sir.
Not only you are limiting the primer concentration, but there could also be changes because of the amplification efficiency.
Yes, sir. Definitely it can happen. So ultimately by changing primer concentration also we are changing only efficiency.
Yeah.
So by look actually it is same, but the wells are deep well actually. So it can accommodate high profile tube. So because we are like taking 40 microliter volume at the end actually for PCR. So when you are adding 40 microliter, so full 40 microliter should be accommodated in the normal PCR. If your whole tube is not inserting in the block, in that case upper layer of the droplet will incubated at proper temperature in comparison to lower level. So in that way your amplification will not be in proper condition. Okay, if you using deep bell, then your whole tube is going inside the block, so all droplets are facing same temperature, or they are being incubated at same temperature. So like in deep bell block you are providing better uniformity for your droplets. So Akansha can give this answer.
Negotiable.
Yeah.
You want to buy tomorrow, she will come to you today.
So Akansha.
The question is very interesting, as if he is going to buy tomorrow.
Yeah.
So in a lighter way, okay, she will tell about that.
So, basic cost in the ballpark figure of the system lands down to up to like 70-75 lakhs.
That's all.
Okay.
He's already switching.
No, it is negotiable.
It's negotiable.
Of course, that is the ballpark figure. You let me know when you're planning to buy it.
Exactly. Yeah, by the way, he's a scientist also in ICR system. And, probably he may buy. Okay, good. This is a new technology. The purpose of bringing this lecture here is you should know what is going on. I was so surprised to see that because Dr. PCR is right in 1992 itself. 95. That means things are going on in the background. We are not aware what's going on. Very nice lecture, sir. So,
Thank you, sir.
then again, innovation. That is the beauty of it. And it's all about something new. Any more questions?
target, copy number of my target is more than 20,000. How can I continue?
So suppose you are adding too much DNA, okay? So you know the concentration of DNA which you are adding in your reaction, okay? But suppose you are using a machine or any gadget, actually any instrument, a small instrument, through which you are quantifying, and that is not giving you accurate quantification. So in that case, what will happen? By mistake you will add more DNA. Suppose it is underestimating your reaction. Suppose you are using normal nano drop or something, and it is underestimating your reaction, and it is giving you lesser concentration. So you are adjusting that concentration in ddPCR. Okay. So you are adding more. In that case, what will happen? You are saturating all the droplets. So until you saturate all the droplets, what will happen? You are able to quantify. The software is able to quantify. But suppose if there are no negative droplets, at least ideally there should be at least 10% negative droplets. So suppose out of 20,000, at least 2,000 droplets should be negative. Okay. So that's why I said around within the range of 10 to the power 5 you can play. Okay. So 1 copy to 10 to the power 5 copies can be used within this reaction. If you are adding too much, like more than that, then you are saturating all droplets. So I just want to show one slide actually. With that you will understand. I am just searching that. So with that single slide actually you can understand in a better way, but I don't know where is that slide.
Sir, I have a
Yes,
couple
sir.
of other small questions.
Yeah, sir. Keep on asking.
You told you are making the droplet.
Yes, sir.
Partitioning.
Mm-hmm.
In the process of doing so, whether the DNA, any damage will happen to the long DNA strands?
Any damage? So, like whatever DNA damage,
Big genome.
any big genome, okay, sir, due to partitioning.
Intactness if it is lost.
So here we are not doing any shaking or something, okay. Only just through capillaries, sample is being aspirated, okay. No agitation. So during aspiration also, if that DNA can break, that cannot be controlled. It is very difficult because it is in cartridge, the sample is being aspirated through the vacuum, okay. So on the other end, vacuum is being created, so the sample is migrating to other end, okay. And even with that much pressure, if DNA can break, that cannot be controlled.
Okay. Maybe very negligible.
Yeah, very negligible kind. So if you have any like doubt in your mind, you can ask. Okay, don't think that any question is. So digital means like.
Listen to this. Sir is asking why the digital is prefix.
So digital means like it makes our life easy. Okay. So like you can directly count in numbers. Okay. You are not doing any background calculation. So digital means whatever you are doing in partitions, and in partitions also, after doing all the background work, it is giving you direct number. So direct number means if someone is giving you some data, and in that data you have to do some calculation, then only you are getting your final number, it becomes difficult. Okay. So while in this ddPCR actually it is doing all the process in partition and other things, and finally you are giving you number in the form of copies. Directly digital number it is giving. Okay, exact number it is giving. I can show you one slide also regarding that.
Digital balance.
So yeah, so the chances of error are like less actually in that case. So whatever number, exact number is there, that is being shown here. I'm surprised that is not here in this. This slide. So suppose when you are, this is example of sample dilution, okay, that how much sample you should use, okay. So suppose you are quantifying your DNA or RNA before doing ddPCR, and then you put your DNA in the reaction, okay. So suppose after quantification you realize that your sample is very much diluted, okay. So in a 20 microliter reaction, 10 microliter in 11 microliter, all the other components will be there, and remaining 9 microliter you can add your DNA or RNA, okay. So like whatever supermix you are using, that is 2x supermix. So in 2x supermix, 10 microliter will be supermix. In 1 microliter you can add primer and probe, and remaining 9 microliter you can use your DNA. Suppose you are using 5x supermix, then in that case only 4 microliter of supermix is required, and remaining you can add your primer and DNA. So that way you can increase the concentration or increase the amount of input material, okay. So even then, suppose whatever quantification you have done, on the basis of that you have added DNA, and the DNA concentration is very less. In that case you will see only few number of positive droplets in upper side and more number of negative droplets. If you increase the concentration of the DNA, okay, so what will happen? Number of positive droplet will increase, and chances of having more number of copies in a single droplet are also increasing, okay. If you add more amount of DNA, in that case you will see more number of positive droplets are there and less number of negatives are there. If you increase it in very high concentration, you are seeing a good cluster of positive and very less negatives, okay. And if you go up to saturation level, so in that case only positives are there, there are no negatives, okay. So when this kind of condition is reaching, means you are adding too much DNA in your reaction, so you are saturating your reaction. So when you reach up to this level, means software cannot quantify sample accurately, okay. So in that case, suppose in first reaction itself or first experiment itself your reaction is coming saturated, then better you do 10 times or 100 times dilution, and then you do further experiment. So this way actually you can decide. So suppose if you don't know that what is the concentration of copies in your sample, in that case you do serial dilution series in first experiment itself, otherwise you run experiment and you just see the data, and on the basis of that actually you can decide in further runs that how much DNA you should use. So if it is saturating, means you are adding too much DNA, or the techniques on which you are relying for quantification, initial quantification, that is not working fine. So this problem we have faced at many labs. They are using any normal instrument actually for quantification of DNA or RNA, and with that the instrument is underestimating the sample, and they are adding it to reaction, it is coming saturated, and they say that there is nothing is coming in the reaction. So when you do dilution, everything becomes perfect actually. So we don't need too much DNA here actually, we need less amount of DNA. So just for example, actually for human DNA, I can tell you how much DNA actually can be used for application, different applications. So if application guide is there, a PDF. So you can also download this application guide from Bio-Rad website. Is it here in this folder? No. Oof. So that actually one mutation guide is there, or rare event guide, in which you can, that is not in this pen drive, okay. But actually on Bio-Rad website itself you can see there is one guide available. So in that actually it is clearly shown that for different type of DNA how much amount you should add. Okay, so with that actually you can take help that if my sample is the genome size is small, so in that case how much concentration I should use actually. So lot of literature is available on website, lot of literature, or like if you provide me your email ID I can send you on mail. So
I think we can discuss continuously like this. There is no limits for that, but somewhere you have to stop. I think Prashant has given an excellent presentation about the PCR, new technology. Now, your imagination and you have to find the reason for using this kind of technologies. I'm sure there are a lot of advantages. Already 85 installations are there in our factory, and top end medical institutions are using this. Certainly, it has got its own benefit over other technologies. That is for sure, because we don't have to have a calibration curve. So for absolute quantitation, DDPCR is one of the fantastic technologies. There is no doubt about that. And he has given a kind of overview of this technology that is going to be more and more popular now, because more and more genomics data, metagenomics data, everything is there in the public domain. People would be using it for their own research program validation and so many other applications. So thank you very much, sir, for taking out your time and addressing our students. And this program is going on for the last 9 days. Today is 9th day. Tomorrow is the validation. And the students were exposed to a variety of genomic technologies, bioinformatic pipelines, wet lab experiments, and we thought a new technology like ddPCR would add up to the, it is something like an icing on the cake. So thank you so much, and thank you for the team from Bio-Rad for arranging this lecture. It's amazing. Thank you very much. So I will call Dr. Ramakrishnan to honor our speaker. Sir, please come this side, sir.
yes.
Somebody can take a picture.
Oh, okay. So he wants this too. Thank you very much. Thank you. Yeah.
Yes sir, please.
Thank you.
Thank you sir.
Yeah, thank you very much. Thank you.
So I will call upon Akansha Shekhar for coming onto the. Soeb is our own person, so I consider as our own team. Okay, give a big hand to the speaker. Thank you so much.
Thank you everyone, and just
Very interesting questions came. So that's really amazing.
so
Now,
this.
let us break for lunch.
Okay. So if you have any further questions, just mail me.
Exactly.
We will reply.
Kalevanan, please note this email.
Or you can contact Akansha as well.
Yeah, exactly. So, since the technology, its importance, and the innovativeness, we gave the extended time. Thank you very much, sir.
Thank you, sir, for
taking
giving
your
actually
time.
allowing too much.
Thank you.
Yeah.
No problem. No issues.
Timing, and thank you to all the people sitting here. They were so attentive because the questions which were coming out to us shows that you people
Yeah.
are very attentive, and you were enjoying the technology.
For lunch,
Yeah.
come back by around 2:15. Let us have the next presentation. And the lecture schedule from 12:15, we will shift it to maybe 4 o'clock, maybe 3:45. Let us see how the flow goes in the afternoon.